Good afternoon. I'm Karen Jones, President and Executive Director of the wAIHA Warriors, and I'm truly honored to welcome each of you to the wAIHA Warriors' third annual patient meeting. It is absolutely incredible to see our community grow year after year, and I'm so grateful for your presence here today. Whether you're a patient or a care partner searching for better treatment, mental health support, a friend who understands, or searching for the elusive remission, you're in the right place this weekend. To our researchers, our industry partners, Novartis and Johnson & Johnson, our exhibitors, Recordati and CAD, we can't do this without you. But we also don't want to do this without you. So thank you very much for your support. Every year, at the top of agenda draft number 1, I won't tell you how many there ends up to be, I type 4 words: inform, educate, support, and celebrate, because that's our commitment to you every year during this annual meeting. Over the next 2 days, you'll get information straight from the experts. Our wAIHA Warrior medical advisors, Dr. Panch and Dr. Irina, and industry partners Novartis, Johnson & Johnson will share a patient perspective of clinical trials and some of the latest in wAIHA research. Tomorrow, Dane Christansen of Health and Medicine Council will provide an update on the evolving landscape of rare disease policy and funding, which is so needed in this time of turmoil. And throughout the weekend, we'll hear many patient experiences that will help you understand how your own story fits into the broader community of the wAIHA Warriors' journeys. Education is empowerment, and I know this from coming from many years in public education and academia, but especially when living with a rare disease. We'll learn how to obtain a proclamation in our state with legislative coordinator James Romano of Care and Cure Partners, from patient advisor Eric Alderman's assistant Sean Powell. We'll get insight into a clinical trial and how it may or may not be appropriate for you. Most importantly, we'll learn from each other, because every shared experience is a lesson for all of us. Alone, we cope, but together we support one another and we thrive together. With Dr. Fuhrer, unfortunately, she is ill and she will not be here, but we will be doing something else with Sean Powell and Mallen. We're going to dive into things like anxiety, depression, intimacy, creativity, and how to build our circle of support, which is so necessary when you have a rare disease. Jen Raber is going to lead a panel discussing hope for remission. The wAIHA Warriors are here to support your journey, and every one of us in this room is also here for you. Our strength is in our shared experiences. But how could we possibly celebrate wAIHA? wAIHA is exhausting, it's anxiety-ridden, it's unpredictable condition. So what could there possibly be to celebrate? I'll tell you. Every increase in hemoglobin, every full night of sleep, every walk taken without needing to rest, every day you simply feel better, feel a little bit more like yourself. Feel a little bit more like yourself before wAIHA. We celebrate your quiet resilience, your full-blown meltdowns, your small wins, and the battles that only you see. We celebrate all that you have overcome to be here this weekend, and we don't take that lightly. We know how difficult it can be to be away from your family and your jobs and all of the things that you have at home, to spend your time with us, and we are truly grateful for that. We'll celebrate all that you've overcome to be here, and we celebrate you and your journey, and we'll support you in any possible way that we can. So welcome to the wAIHA Warriors' third annual patient meeting. Thank you.
So, with my pleasure, I'm going to introduce Dr. Sandy Panch, who became a friend during those meetings. Dr. Panch is a great friend, researcher, as well as physicians. She's committed to pursuit of clinical and translational studies in the niche area of hematology and transfusion medicine. And her research, clinical research focuses on evaluating and engineering blood cells for improved transfusion, transplantation, and immunotherapies. She's fortunate to be able to see a focus of patients with ITP, as well as warm autoimmune hemolytic anemia and cold agglutinin disease in Seattle and greater Washington area. And she also receives referrals from Wyoming, Alaska, Montana, Hawaii, Oregon, California, and Canadian Pacific Northwest. So I don't know how you do that. She also serves as a medical advisor, board director, and member for patient-led organizations, including our own wAIHA Warriors, the CAD Foundation, and the PDSA. And I want to welcome Dr. Panch.
Thank you. Thank you, everyone, for having me back here this year. And it's truly an honor to be amongst all of you. I always, feel like I go back learning more from you than I have to give. And so I was going to share, this is a nerdy talk, but I'm going to try and, talk, or I should say, we can make it as interactive as possible here, and feel free to stop me and ask questions. I might walk around a little bit, so you might hear me going in and out. Okay. Oh. I think it's a small enough room, and I have a loud enough voice, so. Oh, I see. Okay. Okay, so then I can't walk around too much. Okay, that's fine. All right. Okay, so I'll talk about furthering frontiers in red blood cell autoimmunity, as well as alloimmunization, which I'll talk about in the second part of my slides. And, we do studies either starting from the patient and then take it back to the bench or to the lab research side, or sometimes we start from the bench and bring it to the patient side to see what works, okay? So I will talk about the opposite direction, because I see patients and then I take samples back and I study them to understand disease better. Okay, this is our land acknowledgement at the Fred Hutch. These are my disclosures. I will also talk about some drugs off-label. Acknowledgements, I'll say, as Dr. Murakhovskaya mentioned, I study engineering blood cells for improving transplantation, transfusion, as well as immunotherapies. And initially, I studied stem cells as a fellow and then did some immunotherapies. And most recently, I study responsiveness to transfusions. And as many of you in the room know, autoimmune hemolytic anemia is one of the conditions where people aren't responsive as well to transfusions. But there's also other complications, which we'll talk about. So I'll start with autoimmune red cell destruction, wAIHA, and I'll share a little bit about the CAD research that we're doing as well. So we'll start with immune profiling patients with wAIHA, okay? So autoimmune hemolytic anemia, I'll start with talking a little bit about differences between phenotype and genotype, okay? Because I'm trying to make a case here for how our studies connect phenotype, patient phenotype, and genotype. So the phenotype is basically what you present with clinically, okay? Which means that in wAIHA, you present with anemia or jaundice or low hemoglobin, makes you feel fatigued, quality of life is clearly reduced. All of this is your phenotype, okay? Physical form and biochemical pathophysiologic processes are also part of the phenotype, okay? And so the cartoon on the right here shows you differences between warm and cold autoimmune hemolytic anemia, but specifically talks about the phenotype, which is, how high is your LDH or how low is your haptoglobin, are you more yellow than in other cases, so on and so forth, okay? So we all in this room have experience with, discussing pathophysiology of warm or cold autoimmune hemolytic anemia. There's a whole slew of mechanisms that have been studied. Historically, people thought it was predominantly autoantibody mediated. There were antibodies against your own red cells that were chewing it up. Increasingly, we recognize the role of other white blood cells like T lymphocytes or B lymphocytes, NK cells, macrophages, which are these types of cells that chew up these red blood cells once the antibodies are bound to them, which reside in the spleen and the liver and other organs. And so over time, there have been a slew of different agents, drugs that target these various pathways, right? And so the big question for us clinically is when a patient comes to me and says, well, yeah, you gave me 6 different options, which one should I pick, right? So I don't know the answer to that oftentimes. I give them the pros and cons of various drugs. In fact, I just exchanged a story with a colleague, a friend, and a patient. And, we were talking about, well, it's great that worked for you, but I have a hard time convincing patients that that's the right drug for them because each one's body is different. The way you react to these drugs is different, right? So we start with asking the question, can we connect the phenotype, okay, to anything that might be available, whether it's the genes or anything in between, People often ask, does stress affect wAIHA or CAD? Does what I eat affect it? Does how much I sleep affect it, right? We don't know the answers to these. And we think that these answers probably lie somewhere between your genes, which is what the genotype is, and the phenotype, which is what you present as. I mean, everybody presents with anemia, but we know that the underlying pathways may be different across different people who have these symptoms. And so our primary question here was, can we connect or can we look at the person's phenotype and see if we can connect it to their genotype and what are the things that lie along the path, okay? And if there are environmental influences or stressors, which particular paths do they affect, okay? So to begin with, we ask the question, let's start all the way with the basics, which is the genes, Are there specific genes or is there one single gene that we can identify? This is a question that's come up before. Can you link one single gene that has turned awry in warm or cold autoimmune hemolytic anemia, which then results in all these downstream things that are happening, right? That's the question. We don't know no, probably not, but I'll give you some data on that. So in pediatric patients, okay, these are rare diseases, they're not very common. There are some pediatric disorders which are monogenic, which means that there's one single gene that has gone awry, which can explain their large spleen, their large lymph nodes, and several of them also have autoimmune problems, including warm autoimmune hemolytic anemia or ITP or other conditions, okay? This is in a very small subset, typically in pediatric patients, okay? And so what we found that in adults, we know that 50% of warm autoimmune hemolytic anemia is idiopathic, which means we don't know the cause for it, okay? So we did a study at our institution looking at patients with Evans syndrome and ITP. Evans syndrome has warm or cold and ITP, or it could be autoimmune neutropenia. But we looked at these patients and said, well, is there a single gene that can cause this? And the answer we found was no. We looked at all of their genes, specific genes, I should say, not all of their whole genome. These days we can sequence the whole genome, but it's still quite expensive and it's cumbersome for somebody reading it. So we pick out targets. And we did a targeted sequencing, and we didn't find answers to these particular cases of wAIHA or Evans syndrome and link it to these particular genes, okay? But what we did find was almost 50% of them either had a personal history or a family history of somebody, other family members having autoimmune disease, okay? So there's clearly something in the genes. It may not be that one single gene, but it must be something in the genes, right? But how do we study that? And so if you go back to your high school biology, and I'll make an analogy here of, how does one make chocolate chip cookies from grandma's recipe book. So if you think about the DNA and transcription and translation from high school, right? The DNA molecule is like grandma's cookbook, basically. It has all the recipes under the sun in excruciating detail, okay? But what you want to do really is make chocolate chip cookie. So you say, well, guess what? I am going to take just the recipe for the chocolate chip cookie and transcribe it onto a paper so it's easier for me to not have to keep looking through all pages. So that process of transcription is what happens from the DNA to the mRNA molecule, okay? And then the next step is actually making the cookie from the recipe. So that process is called translation, which means that the mRNA molecule calls in ribosomal RNAs, which bring in all the ingredients, and the process of making protein, or the chocolate chip cookie here, from the recipe book is called translation. Okay, so there's DNA, there's transcription to mRNA, and there's translation to protein. And the DNA can sometimes be affected by environmental factors, or what we eat, or how stressed we are. But all of these pathways along the way are far more susceptible to environmental insults. The DNA typically is very strong, okay? Because if you mess up the basic code, you can mess up many things downstream, okay? But expression of these genes can often be affected by various environmental insults or medications we take and so forth, okay? So that's basics here. So what we decided to do was not just look at the genes or the genotype and compare it and correlate it with the phenotype, or why some people have anemias versus not, or jaundice. We said, let's look at all the pathways in between. Let's look at the transcription process. The output of transcription is what we call transcriptomics. The study of transcription is transcriptomics, and proteomics, and cytokine analysis. So we did what we call a multi-omic approach and studied all the steps, tried to dissect each step and studied each step little by little to see if we could figure out the connection, because it seemed like a single gene was not the answer to our question, okay? So we did this as part of a multi-institutional study. It was a retrospective study, and we did it in the context of a drug called fostamatinib. As you all know, there's no approved drugs, FDA-approved drugs for wAIHA. And so fostamatinib came close, actually, to be approved. And Dr. Irina and others were part of the original study, the randomized control trial that looked at fostamatinib versus placebo, and found that there was a 36% response rate in these patients with the use of the drug. But unfortunately, there were discrepancies between data from Eastern Europe and the rest of the global sites. So it didn't meet primary endpoint, or it failed to achieve the outcomes that they expected, and so the FDA did not approve the drug. But several of us thought that it did work in a subset of our patients. And so we got together, a group of us, and said, well, let's ask that question in the real world setting and see if it works. Okay, so that's what we did to start with. And we screened about 100 patients across 6 sites, and we found 22 patients who were not previously on the fostamatinib trial were enrolled. And we looked at primary outcomes, which means how much did the hemoglobin increase? Okay, that's the main thing we are interested in. But we also looked at, were they able to come off steroids? Were they able to get less transfusions? Did they reduce their ER visits? Okay, so we looked at all of these things, and of course we looked at side effects of the drug. But in a subset of patients, we did this whole multi-omics profiling that I talked to you about. We had patients who were enrolled, whose charts we reviewed, but we also had sample on them, so we were able to look at their DNA, we looked at their RNA, we looked at their protein, cytokines, all of these things, okay? And so interestingly, the response rates, the initial response rates were as high as, well, I'll show you that in the next slide, but 60% of them had wAIHA, and another 40% had Evans syndrome in our group, okay? And several of them had at least 3 treatments, and this is excluding splenectomy. So they were a multi-refractory group, okay? There were some adverse events, not significant, nothing that was grade 3 where patients required significant hospitalization, so forth. But this is the data I wanted to show you. 64% overall, and this was multi-site. So we aggregated all the data, and about 64%, a little more than a coin toss, I would say. So it's not great, but it's not bad either. 64% responded. Yeah?
You have to use the microphone.
Sorry. Okay.
We won't be able to hear you.
Yeah? No, that's great. Okay. Can you hear me now?
You can just take it out of the holder.
Okay. Okay, that's perfect. Okay. All right, so 64% responded. That's good. Okay. And you can see differences. You know, the blue line there is the hemoglobin in the responders. The red line is the hemoglobin in the non-responders in the first two figures, A and B there. Change in hemoglobin and hemoglobin. C and D are basically the LDH, the retic count. The LDH retic count trends were not great. It was retrospective. You couldn't really see, and a lot of patients were on different therapies. But we looked at FOSTA patients 3 months before and 6 months after. Overall, you can see a nice separation in responses, hemoglobin responses between the responders and the non-responders, which was reassuring. So then we asked the question, well, in our subset, can we look at the genes? And it turns out, just like in our original cohort of Evans syndrome patients, we didn't really see a difference in the inborn errors of immunity genes between the responders and the non-responders. That's what I show you there on the left, wAIHA, okay, that's the wAIHA group. On the right are controls. And controls were age and sex matched, and it looked like they both had an equal number of, excuse me, what we call pathogenic genes, increased risk genes, variants of uncertain significance, and so forth. We also looked at their bone marrow biopsies in about 15 patients, 15 out of the 22 patients, and there were some patients who had variants of uncertain significance, but it didn't look like it was different between the responders and the non-responders, okay? So all this to say, I don't think the genes were giving us the answers, okay? So we then looked at the next layer. We found some interesting information here because we looked at cytokines, we looked at gene expression, which was what you find after you transcribe the recipe, right? So we looked at gene expression, and there were quite a few genes that were expressed differently between the responders and the non-responders, which means there were environmental influences, dietary influences, other things that influenced how these genes become proteins, okay? And so we found differences in certain pathways, like JAK-STAT pathways and NF-kappa B pathways and macrophage migration inhibitory factor levels, okay? And everything suggested overall that inflammatory pathways were higher in the non-responders, and the inflammation seemed to be more suppressed in the responders overall, okay? Same thing with the single-cell RNA sequencing. That also gives you more information on, at a cellular level, whether there are differences. And again, it seemed like there were differences in subsets between the various WBCs in these patients, and it looked like there were more inflammatory types of cells in the non-responders than in the responders, okay? So overall, like I said, there was a 60% response with the drug. There were adverse events, okay? Overall, we didn't find any differences in gene mutations per se, but what we did find was differences in cytokine expression, in gene expression profiling or transcriptomics, and so forth. Now, just to remind you again, these are not tests we routinely do in the clinical lab. So we couldn't, at this stage, tell you, hey, your macrophage migration inhibitory factor is low. If I give you the drug, fostamatinib may be a good drug for you, right? So we're not there yet, but that's where we want to head, okay? And so that's what we asked. We said, now that we know it's feasible, can we extrapolate this information and ask the same question with regard to all the drugs that we have out there in the context of use for wAIHA and CAD and ITP, right? And so can we go from the current state where we just sequence therapies, one after the other, clinical trial after trial after trial? Can we say, the future of medicine should be more personalized diagnostics? Can we say, I will do cytokine analysis on you and transcriptomics, and I will tell you which is your poison, okay? So coming back to this figure of all the cells that are awry and all the treatments that have been sequenced and developed, right? We want to ask, can there be biomarkers that we can develop along the way, okay? And this is not my idea, and it's not novel, okay? I'll say that in full disclosure. People have been doing this since the 1960s, okay? And that's the data I show you here. People have looked at cytokines, at cell populations, at various subsets. They've also correlated it with age and steroid response and things like that to decide who might or might not respond to splenectomies, or who might or might not respond to steroids and IVIG and so forth, okay? So there are some cytokines that were identified. Now remember, these are all studies in very small populations. So my husband is in tech, so he always says, well, your data, your p-values are like 0.04 and you jump for joy, Well, that's because I have 10 patients. You have, I don't know, reams and reams of data, right? wAIHA is a rare disease, CAD is a rare disease. If I get 10 patients to analyze, it's a pretty big deal, right? But it's definitely the caveat with all of these that it's a rare disease, so you don't get as much data, okay? So then we ask the question with what we have. With splenectomies, several of you in this room have undergone splenectomies, right? And so wouldn't you wish that there was a test that would tell you up front whether or not you would be a responder or not, right? There is something. It turns out it's not great, though, but there is something, okay? And I'll tell you the caveats, okay? Because, it was done in a very small, or it was done in multiple small groups, okay? So there is radio-labeled scintigraphy, okay? What that is they take your own red blood cells or your platelets, depending on whether you have ITP or wAIHA. They radio-label it, which means they add a marker, and they infuse it into your body, and they see where the destruction happens. After you get your own red cells that are tagged, they see if it goes predominantly to the spleen or to the liver. If they find that you're mainly destroying it in the spleen, they image you after they give you your own red cells. If it's all being destroyed in the spleen, they say, well, I think you're going to have success with splenectomy, okay? Versus if it's mainly happening in the liver, the likelihood that you would not respond to splenectomy is higher, okay? But it turns out that handling radioactive substances in a regular lab is a huge regulatory burden, okay? Because there's other patient samples that are coming in and out. They don't want to mix radioactive samples. They don't, you yourself are being exposed to radioactive agents, not much, small quantities. So it's very cumbersome to do. And you don't get, the sensitivities on a recent ITP study was 90%, which was pretty good. If you have a 90% chance that it can rule out whether or not you would respond to a splenectomy, I think that's pretty good. Sometimes we joke, we tell our patients to take a trip to Europe if they're thinking about a splenectomy, because in the US, the insurance doesn't cover radio-labeled studies. But there are centers in Spain that do it, not in spleen, but in Spain that do it. Okay? So I think about the spleen all the time. So much more than I talk about Spain. Okay? So, it is an option, okay? So sometimes I tell my patients to take a trip to Europe and see if something like this could be done, okay? I don't think it works as well for wAIHA as it does for ITP, though, okay? So the next drug, daratumumab. Guys, I had somebody in this room that actually advocated it, which I'm very happy to hear, because I do have patients who I am talking to about the drug. I'm not advocating for it. We're just thinking about all the options. So daratumumab is a plasma cell depleting drug, okay? It's a CD38 monoclonal antibody, just like rituximab, which acts on CD20. This acts on CD38. Now, we give it for a condition called multiple myeloma, okay? But it's interesting also that the CD38 antigen is not just expressed on plasma cells, it's also expressed on other cells, T cells, B cells, NK cells, okay? So there was one study, very small, that looked at CD38 expression on T cells and whether that had anything to do with responses to daratumumab. And it looked like if you were at about 30%, the chance that you would respond to the drug is high. And then there was, again, very small numbers, so take everything with a large chunk of salt here. But I do measure now T cell CD38. So this is not a standard test either. We ask our hematopathologist to look at it and see. There was one patient on their study whose recurrence of wAIHA actually corresponded with increase again in CD38 T cells. That's what is shown there, okay? And so maybe that's an option. In fact, this is a slide of a patient who I gave daratumumab to with refractory ITP 6 months post-COVID. She got ITP after COVID vaccine, unfortunately. You can see there in the bottom, very, very low platelet count. She didn't respond to about 7 different therapies. And then I gave her daratumumab, and look at what happens to her. platelet counts, the pink line there. We also measured, I do the same thing, transcriptomics, proteomics, cytokine analysis. It looked again like her inflammation was really subsiding after the DARA doses. Okay? Now I will say this, I don't know if that was just because she was 6 months out of the vaccine and the vaccine effect wore off, or if it was actually the drug. Okay, so I don't want to give it too much credit just yet. We know that from early studies, there's about a 50-60% response rate with DARA. You still have to think, though, that what works for one person may or may not work for everybody. Okay, fostamatinib, I showed you the data already. It looks like there are some markers that we could use. I'll talk a little bit again about those pediatric conditions that I told you about right in the beginning, where there were monogenic or single gene disorders that resulted in autoimmune cytopenias, ITP, wAIHA, okay? And so there are targeted drugs. Leniolisib is one of them. Abatacept is another. These are all targeted drugs that can be used for these conditions. And the question is, can you revisit those pathways and see in your wAIHA patients who don't have the gene mutations but have pathway overexpression or activation, whether you could repurpose some of these drugs and give it to patients who don't have the gene mutation but have pathway activation. Okay? So my conclusions are there are things that we can do, they're not great, but we should start thinking along these lines because now we have a lot of options. Okay? The goal is to now start thinking about, well, how do we decide what's the right drug for who? Okay? So we have a registry that we're trying to build here, again, to take wAIHA from a sledgehammer approach to more of a precision therapy, which is the goal. And along these lines, Dr. Crystal Hudson, who unfortunately couldn't be here with us, is a close collaborator with all of us, Dr. Irina and all of us, and we're also trying to evaluate potential drugs that could be used. I'll briefly mention the study we're doing with cold agglutinin disease, okay? Now, I don't know how many of you have experienced this in your clinic or in your lab, but unlike ITP, thankfully we have a test that is pretty good for wAIHA and CAD, which is the Coombs test, exactly, the DAT, okay? But I cannot begin to tell you how often patients come to me who have been diagnosed with, as a warm if they're a cold or a cold if they're a warm, or they just have a positive DAT because they just got IVIG or, for other reasons, right? And so I always go back and ask the blood bank, well, how strong is the DAT? Is it 1+? Because they just got IVIG, or is it 4+? Okay, they don't always report it, so you have to ask. And then you have to ask the second important question, which is it IgG or C3? Because typically colds are complement mediated and warm autoimmune hemolytic anemia is immune globulin G mediated, okay? And so you do want to know the difference. Most patients who are referred just get treated with steroids. Steroids don't work as well for colds, okay? And they work for warms, but you're not supposed to keep them on it indefinitely either, right? Rituximab works for both, so you still don't know what it is. So most community physicians will say, give steroids, give Rituxan, and then they don't know what's going on, so they refer, right? And then you start asking questions from the basics. Is it a warm? Is it a cold? Or is it mixed? So when we did this, ironically, even within the UW lab, we sent it to the first lab. I had a 99% suspicion that this was actually going to be a cold autoimmune case of cold autoimmune disease, but all their testing kept coming back negative. And then we sent it to a different lab within our system, to a reference lab, and the tests actually came back positive. So my suspicion that they had cold agglutinin disease was actually right. And we tested about 16 patients, and about 11 of them were falsely negative initially, and then they were positive. By repeat testing, it turns out that one of the labs transported the samples. The ones that did it right transported the sample warm and tested it warm, which is what one needs to do for cold agglutinin disease. Okay, whereas the other lab was collecting it at room temperature, storing it, sitting on it, and then re-warming it, but that didn't help the antibodies that were bound. They were just destroying the red cells and they were gone, okay? And so that was an important lesson we learned in terms of testing and the importance of all of the results that we get from these tests, okay? So I'm going to switch gears now a little bit. How much time do I have? 17 minutes. Oh, nice. Okay. Okay. So I'm going to, this is a story I'll tell you. It may not be directly applicable to your situation, but I want to share the story as a cautionary tale. It's also pretty dramatic. Several of us lost a lot of sleep for many days. This was a patient who needed red blood cell transfusions, and she was refractory or non-responsive to these transfusions because of underlying alloimmunization, which is different from autoimmunization. Autoimmunity is where you make antibodies against your own red cells. Alloimmunity or alloimmunization is where you make antibodies against transfused red cells, which you can develop if you have autoimmune hemolytic anemia because you do get transfused. In fact, one of the big conundrums in transfusion medicine is the inability to detect underlying alloantibodies in the presence of strong autoantibodies. Okay, that's why we're so worried about giving people with autoimmune hemolytic anemia transfusions, because you may develop antibodies. Not your own, but antibodies against transfused red blood cells, especially in ethnic minorities, okay? Because the donor pool is predominantly white, let's say, and the recipient is of an ethnic minority, it can impact it, and I will show you that data. So, we called it, was it a red herring or killer whale? And I'll explain why we said killer whale. But this was a case of a near fatal immune hemolytic anemia, okay? So I'll talk about the clinical case, the lab evaluations, management, and what we learned. She was 63. She had one child, or one adult daughter, originally from the Philippines. She was admitted for an elective cholecystectomy, where they take out your gallbladder, okay? And she had gangrenous cholecystitis, so she had to be on antibiotics. Post-operatively, she had the surgery, and then her hemoglobin dropped from 10 to 6, okay? So they said, whoa, she's bleeding a lot, okay? And so they gave her 6 units of red cells, okay? And previously, she had a non-specific positive cold antibodies. When you go in to get blood, they always do what is called a type and screen before they give you blood. So they look at your blood type, where they look at your ABO blood type, and they look at your RH blood type. So when you say I'm A positive, it means you are blood group A, and your RH type is positive or negative, it's positive. But there's about 360 other red cell antigens that you do not type for on a regular basis because it's impossible to match, okay? And you just assume that the donor pool is homogeneous with your blood type, and you hope that it doesn't work. Now, I'll take you back in time to the 1600s, okay? People would transfuse xenogeneic blood, which means sometimes they would give patients calf's blood, soothing calf's blood, and hope that that would help calm their syphilitic frenzies. That actually originally caused people to ban, the Parliament of Paris to ban transfusion forever. It lasted 200 years after that because the real transfusions, human-to-human transfusions, started in the 1900s after they identified ABO blood groups, and they were able to show that people can do cross-compatibility testing, which means you take the patient's red cells, add the donor's antibodies or vice versa, and show that they match. And then they transfuse. That's what the type and screen is. You type the patient, but you also screen to make sure that there's no incompatibility, okay? So we've come a long way since the 1600s. But this patient had incompatibility, but we didn't know what to make of it. Sometimes you have these weird signals, and you say, well, I don't know what that is. She just lost a lot of blood, we gave her a bunch of blood, and now she has this weird incompatibility signal. But guess what happens? She really drops. We gave her 6 units, she holds on to it for about a day or so, and then she really drops again to the 6s. And now her DAT has become really strong. Her C3, which is indicative of a possible cold, is now 3+. Her IgG is 1+, and overall she's floridly positive. And when you look at her blood tubes, it looks like she may have hemolysis, just like we can see when your urine is dark in autoimmune hemolytic anemia, right? You can have that with alloimmune hemolysis as well, but we didn't know that at that time. So we sent off the antibody test. This is to see, well, she may have an autoimmune hemolytic anemia. At this point, we're thinking she probably developed autoimmune. You know how after stressors, including surgery, there are several patients who develop autoimmune hemolytic. So that's what we thought she had. Because we started her on steroids, we gave her more blood, and we said, well, this seems like a weird autoimmune hemolytic anemia that seems to have been triggered after a surgery. So we sent off testing to the blood bank. We said, just in case this is something else. They said, we don't know what this is. We're finding signals for an antibody of undetermined specificity, which means we can't find an antibody against the ABO group or the RH group or the transfused Kell, Kid, Duffy. These are all blood groups that we don't routinely test for. They do test for it, and they say, well, we can't see anything. So that's her urine, guys, by the way, on the side there, and her plasma. You can see that it's getting dark. And there was an important clue in her story. You know, autoimmune hemolytic anemia patients, typically when you transfuse them, at least for a few days, they say, well, I actually feel pretty good with the transfusion, right? But this lady, you know, she was lying in her bed and she said, I feel like death every time you guys give me blood. Okay, so that was huge. And this is what took us back to the 1600s, because if you went back and read those textbooks, they would say, patients will tell you they feel like death when you give them the wrong blood. And by that, they mean they gave them calf's blood, okay, and there was incompatibility. And then they would go on to describing, well, he made urine the color of chimney soot, okay, because there was just incompatibility and they were breaking everything down and you were killing their kidneys. So unfortunately, that's what we were doing to her, but we couldn't figure out why she was breaking down red cells. Okay, so we said, well, what is this? Is it warm? Is it cold? Is it paroxysmal cold? You know, sometimes with infections, you can have these colds which are paroxysmal. Is it drug-induced? Sometimes there can be drugs that bind to red cells and behave like warms or colds, right? And then we said, well, is it alloimmune? But we're not finding those antibodies. Sometimes infections, we said, well, is it Clostridium-mediated hemolysis? Sometimes there are bacterial infections that can cause blood cells to break down. So we asked whether it was that, and nothing seemed to be clicking here. But she kept dropping. You can see there, significant drops. Every time we transfused her, she would go back up and then drop immediately and feel like death each time, okay? But we didn't have answers. So we looked through everything in the textbook. That's what this shows. Is it warm? Is it cold? Why is it or why not? You know, we worked it all up. And then eventually we said, well, this could be drug or this could be alloimmune because we're not finding anything consistent with autoimmune hemolytic anemia. The story just isn't matching. And so we said, okay, let's look at this more. We went back and looked at the antibody screen. We didn't find anything. We went back and looked at whether she could have other abnormalities that we could identify on genotyping. And I'll tell you a little bit about that. So we did two things. We went back, looked at her antibodies, and then we said, let's genotype her RBCs. Because genes can be picked up. If you have a RH or a Kell or a Kid, you can pick it up on genotyping and say, I know which type she has, and we can match for it. In fact, autoimmune hemolytic anemia patients, we often say, please give phenotypically matched blood, which means you don't just match for ABO and RH, you match for a few other antigens that are common, okay? And so we did that. The serology testing just says, well, it looks like she has an autoantibody, okay? The problem with autoantibody-mediated hemolytic anemia, like I said, is you have to take out layer by layer all the autoantibodies before you start looking for underlying alloantibodies if you had, in fact, made something, which was impossible in her. This process of eluting out all the autoantibodies takes about 8 hours, typically, with techs in the blood bank. It took us 3 days, and we still didn't find an underlying allo. Okay, and we sent it to the Canadian Blood Services, to New York Blood Center. We didn't find answers. So then we said, let's genotype her. This method of genotyping, it's called the array-based genotyping. And I want to tell you guys about this because no test is 100%, okay? Just because you have a test that's negative, it doesn't mean, you know, it's ruled out. So this genotyping array basically picks out particular gene markers and only tests for those, which means if it's ABO, they only test a particular part of the ABO. They don't test for the whole ABO gene, okay? The most common part of the ABO gene is what they look for because it's very, very cumbersome to sequence your whole gene. people just pick bits that are most common. Again, you're threatened by statistics here to say, okay, or probabilities, I should say. Okay, so that's what we did. We didn't find answers. We said, well, I guess it's time to stop transfusing her. So what do? We gave her rituximab, we gave her glucocorticoids, we gave her IVIG, we even started sutimlimab in her, okay, because we said, let's block complement and see if that's helping. But we still don't have an answer for why she's hemolyzing. So finally, someone has a brainwave saying, guys, you didn't look at her whole gene. It's impossible to get all the answers. She's Southeast Asian. She may have gotten blood from people who were not from the same ethnicity, okay? So you need to look further. And so we did something called long-read sequencing. This is a new technology. It's pretty cool, okay? This is the device we used. It's like a little dongle that you keep on your tabletop, basically. In fact, I've gotten pulled over by our regulatory folks because sometimes I keep it on my desk and I have somebody's DNA running there and they're like, what are you doing? That's contamination. You can't just keep it next to your lunchbox, you know? But anyway, so that's what we did. We put in, so what this does is you extract DNA and you run it through this, and it basically reads every base pair in your DNA in parallel, okay? So what used to take like 20 days to do, this instrument does in half an hour. It sequences your whole gene. But that's not the problem. The problem is getting somebody to read it and telling us which base pair means something is off. So there's one guy in all of Seattle, I should say in all of the Pacific Northwest, who knows how to read it. So we ran it, but what were we going to do with the data, right? So we went to this guy. He's backed up like 6 years because he's got so much data to read. And we say, well, push this all the way up your list, you know? We really want answers in this lady because whatever we do is not working. And so that's what he did. This is his fancy analysis. And in 6 hours, he came back to us and said, is she Southeast Asian? You know, we said, oh, wow, okay, I think you're on the right track here. What happened? Well, she may have a really abnormal gene seen in about 0.001% of the population where her KEL gene is not, her KEL antigen is not expressed on the surface of the red cells, even though you detect the gene, because it has something called a stop codon, okay, which means that the gene is there, but there's one base pair which is switched, okay? And because of that, there is what we call a stop codon, which means the gene, remember I showed you that picture of transcription and translation? The gene is there, it just doesn't get transcribed or translated, okay? So the protein, which is the KEL antigen, is not made. So we're like, oh my gosh, this is crazy. Where are we going to go with this? In blood banking, there is one thing we're really afraid of, which is the KEL group antigen, because we don't test for it. We only test for AB or RH. And so there is a saying, an infamous saying, KEL kills, okay? And so the antibody that she had was actually a Keller whale, like I pointed out to you earlier, okay? It was not a red herring. It was not just something that was a distraction, okay? And then we went back to our blood bankers and we said, well, can you look for the KEL null gene or the KEL null antibody specifically? And they said, yeah, we see it. It's there, you know? So, I mean, hindsight is 20/20, right? Now everybody knows it's the KEL gene or the KEL protein that was supposed to be missing because of this, and they went back and redid their methods, and they actually found the antibody that caused it. Anyway, so, and then thankfully, we stopped all transfusions. We gave her sutimlimab, because when all hell breaks loose, sometimes you just have to give them everything under the sun. We don't know what works. We threw the kitchen sink, and thankfully her hemoglobins responded. She's stable now. We just hope that she never has a GI bleed, because there's about 2 donors in all of the US that have the KEL null phenotype, or we have to go to Thailand or the Philippines and bring these donors for her, okay? Interestingly, we tested it in her sister, and her sister has the same KEL null mutation, except that she developed aplastic anemia at some point, so we couldn't get her to donate for the sister. Anyway, so there were lots of learning points from this overall. But I hope I've been able to take you through some immune profiling stories for autoimmune as well as alloimmune transfusion non-responsiveness. Okay, and this is my group. Thank you very much for your time.
All right, good afternoon, everyone. My name is Sean Powell. I'm the Vice President of wAIHA Warriors. For those of you who don't know me, I was diagnosed in 2019, and I'm approaching my 6-year anniversary. So I like to liken wAIHA to being hit with a curveball, and we here in this room are all experts with dealing with curveballs, and we're going to deal with a curveball this afternoon for this presentation, because unfortunately, due to a family situation, Eric was not able to be with us, and we had hoped to show his video and then have a Q&A session with Eric about his experience with the clinical trial and try to debunk myths about clinical trials and get people more interested in them, because that's where our research comes from, and that's where new modalities come into fruition. So we're going to ask that you hold your questions, and that during the reception this afternoon, we will have physicians and our research partners from Novartis there. Please seek them out and talk to them about what's entailed with the clinical trial, how do you get more information, and what the experience is like. So with that, we're going to watch the video with Eric, and then it'll be our next presentation. Thank you.
My name is Eric. I am from New Rochelle, New York. I was first diagnosed about 13 years ago. It was in the winter, and I had a really bad flu, probably the worst flu that I ever had. I just so happened to have a doctor's appointment a week later for my annual physical, and my doctor ran some normal blood tests. And a few days later, he called me, and he sounded very alarmed. And he told me that all of my blood tests were out of whack. He suggested that I go see a hematologist immediately. During this time, I just didn't feel myself. I was really tired, wasn't sleeping well, and it was scary. I didn't know what was going on. I was generally a healthy person. It took really a few months. Once they confirmed at that point that what I had was wAIHA, warm autoimmune hemolytic anemia. The first treatment I had was being on high doses of steroids. It really helped me feel more like myself. And then a couple years later, I was on vacation, and I started to have those really bad symptoms all over again. So we went to all different kinds of other treatments that were out there. I'd feel better for a few months, and I was on this cycle really for a couple of years. And at that point, I really got very frustrated that there had to be something. There had to be something out there that maybe my doctors were missing. And so I went up to Boston. There was a doctor there, a hematologist, who was specializing in warm autoimmune hemolytic anemia, and he was doing a lot of research into it. After I saw him, I did some research, and I saw that some of the clinical trials he was doing were also being done out of a hospital that was only about 30 minutes from here. I decided for a number of reasons to do the clinical trial. Really, probably the most important reason I did it was because I wanted to help myself, but I also feel like I want to help the community. One thing I've learned also about wAIHA is everyone's different. It affects everybody differently. And I thought that being in a clinical trial would, help people who had the same profile like me and the same experience like me. I did have to go periodically to have my blood drawn and blood tests done, and to see her and have other tests done. And that was more frequently than I would have seen her otherwise. For those who might be considering being in a clinical trial, I would say get involved with the patient advocacy groups like the wAIHA Warriors. I'm a member of the group, and we have a lot of resources available that can help you understand what it's like to be in a clinical trial. Being in a clinical trial is your opportunity to be on the forefront of medical science, and for the community, for the wAIHA community, for yourself, that will help all of us in the future having to face this disease.
People living with warm autoimmune hemolytic anemia or other rare disorders face a significant challenge due to limited treatment options. Clinical trials are research studies where doctors, researchers, and people living with a specific condition explore potential new treatments to help us find better ways to fight diseases. A treatment can be a drug, medical device, medical procedure, or a change in a person's behavior, such as diet or exercise. Clinical trials are the worldwide standard for reviewing potential new treatments, a process that has led to advancements in healthcare. They open the door to new discoveries in science and medicine that can help people. By participating in clinical trials, people living with the specific condition can help shape the future through contributing to research. People who participate in clinical trials get regular monitoring, either online or in person, on top of their regular doctor visits. For new treatments to reach their full potential, clinical trials need a diverse group of participants, including people of different races, ethnicities, and sex. This helps researchers understand how treatments affect different people, leading to better treatments for a wider range of people in the future. For certain studies, children under age 18 may also be able to participate, but only with parental consent. Choosing to participate in a clinical trial is a personal decision that everyone makes for themselves. Discuss with your doctor and loved ones to see if it's the right fit for you. If you choose to participate, you'll be joining a group of people committed to medical progress. You'll have important rights and protections for your safety, and your doctor's top priority will always be your well-being. Everyone who joins the study is treated with respect and gratitude for their time and contributions to research. Understanding more about the clinical trial process can help you decide if participating is right for you. All medical research involving people must be approved by an institutional review board, or IRB, which is sometimes called an ethics committee. Before inviting participants to join a clinical trial, researchers, also called study investigators, submit their study to the IRB, where it will be reviewed to confirm whether the study prioritizes participants' rights and safety, and answer specific research questions that will support its goals. Before joining a clinical trial, potential participants receive something called an informed consent form, outlining the most important facts about the study, so they know exactly what's going on. The research team will explain everything: treatments, risks, benefits, and any medical tests that may be involved. Participation is always voluntary. Participants have the right to ask questions at any point and can choose to leave the study at any time for any reason without consequences or impact to regular medical care. Like any medical treatment, clinical trials have potential risks and may have unexpected side effects. Your doctor and the consent form will help explain these possibilities. For example, the new treatment might not work as well as other options or may not work for everyone. It's important to weigh the risks and benefits to decide what is right for you. Clinical trials select people who meet specific requirements. Researchers carefully review people living with a specific disease, along with healthy volunteers when needed, to see if they qualify to join a study. These criteria, called inclusion criteria, might consider factors like a person's age, biological sex, and specific type of disease. Before deciding, you'll also want to think about the day-to-day impact of participating in a clinical trial. Participants may need to consider things like getting to the study location, taking time away from work, and arranging childcare or pet sitters. Your doctor and the researchers will often have guidance and resources available on these topics. During the study, some information about you will be collected, like your medical history and laboratory test results. To ensure your privacy, all collected data will be encoded, which means your information will be identified by a number, not by your name or any details that could link back to you. The group paying for the study is called the sponsor and often covers the costs involved in participation. This might include the new treatment itself, special tests, and even travel to the clinical trial site. Some clinical trials may also pay for your time and participation. Your costs for joining a trial can change based on the state you live in or your insurance coverage. Talk to your doctor about the clinical trial you are considering. There may also be patient advocacy groups that can be a valuable resource to you. Clinical trials have a step-by-step process where researchers carefully review the potential new treatment's safety, side effects, and how well it works. Before examining a new treatment in people, researchers conduct preclinical testing to determine whether it is safe. This is guided by something called Good Laboratory Practices, which is a set of strict rules the U.S. Food and Drug Administration, or FDA, created to ensure preclinical research meets specific standards. After preclinical testing is complete, researchers review the findings and decide whether the treatment should be tested in people. If the decision is yes, the study will move into phase 1 of the clinical trial process and begin enrolling people to participate. The study will start with giving the new treatment to a very small number of participants for a few months to see if it's safe, if it has any unwanted reactions, called side effects, and determine how it should best be used. This is an important part of every treatment's development because it is likely the first time it is being given to patients. If things go well in phase 1, the study moves into phase 2. This may involve a larger group of participants over a longer timeline. Throughout this time, researchers will collect data and participants provide crucial feedback to help understand how the treatment is working. After a successful phase 2, the study will move into phase 3, potentially involving even more participants. During this phase, participants will be randomly assigned to one of 2 groups. The treatment group receives the potential new treatment being studied, and the control group receives either an existing standard treatment or a placebo. A placebo is a substance that may look or taste like the real medicine, but has no therapeutic effect. This is called a randomized controlled clinical trial. Placebos are particularly valuable because they allow researchers to see how well the new treatment works by comparing those who are taking the new treatment and those who are not. Placebos aren't always used alone, though. In some studies, researchers might compare the new treatment to a combination of placebo and an existing treatment. Once phase 3 is complete, the results will be reviewed by the FDA to determine if the new treatment is safe and really works. If they say yes, then the pharmaceutical company is able to share the treatment with doctors who can prescribe it to patients who need it. But even after the treatment is approved and available to all patients, researchers might still keep studying it to monitor how well it works. This is called phase 4. It's also important to remember that not all clinical trials will reach the finish line. There are many reasons for this, but it doesn't mean the work wasn't valuable. Researchers still learn a lot from it, and it can help them develop even better treatments in the future. If you're ever in a clinical trial that ends early, your doctor and your study sponsor will work together to find the best care plan for you going forward. Across every disease, today's treatments are the result of research conducted during clinical trials. Clinical trial participants have shaped many innovations. contributing to our ability to understand and treat certain diseases. If you are considering participating in a clinical trial, talk to your doctor about studies that may be enrolling people diagnosed with wAIHA. To learn more about clinical trials and what to expect if you choose to participate, please visit clinicaltrials.gov.
My name is Jen Raber. I have been on the Patient Advisory Board of the wAIHA Warriors. I'm pleased to introduce Lisa Shea, Director of Global Patient Advocacy Engagement with Johnson & Johnson, as well as Dr. Zia Choudhry, Senior Medical Director of Rare Antibody Diseases, Immunology at Johnson & Johnson.
Hi, everybody. Thank you so much for giving us the opportunity to come and present here. It's been a great conference so far, a lot of good sessions, a lot of good learnings, and being with all of you, especially understanding and connecting with the patients, I think is always rewarding. And great conference so far. So this is a lot of work that J&J is very passionate about. We've always, especially with regards to the autoantibody diseases, we've always been very passionate about learning what the patient experience is. There is a lot of effort in terms of creating specialized research projects. So before I go into this, I want to spend a moment kind of acknowledging the work that happened. It has my name and Lisa's name here, but all of us at J&J have been working very hard in collecting this data. We have a patient engagement research program that we are very proud of. With the focus on rare disease. And Anne Leon, our friend, is here as well from the Global Medical Affairs. She's been a strong component of this as well. So what are we going to present today, right? We've presented at multiple conferences like the NORD and ASH, and some of that work has been captured here in this presentation. We really want to take a moment, take a peek, right, into the experiences of patients with warm autoimmune hemolytic anemia. And who would know better than you guys, right? You live it every day. And we try to learn from you and from some of your peers. We engage them in a robust research fashion and got some great insights, which I think you will find interesting. All right. So conflict of interest, we work for J&J, and we're a part of the organization. So what is wAIHA, right? We've discussed this quite a lot. A rare red blood disease, what's happening, the destruction of red blood cells, it's accelerated. The destroyed red cells are creating, particles which are in the bloodstream, they're going into the tissue, they're causing a wide variety of symptoms. It's very rare, right? Somewhere around 1 to 3 patients in 100,000 people every year. And I was having a chat with Lisa earlier, right? When we talk about research, when we talk about studies, usually the mindset is, the bigger the number, the strength is in the numbers. But when we think rare, given that it's so hard to recruit these patients, even an N of 5 is very important, and that data is very valuable because we can learn so much from it. wAIHA has impact, right? We all kind of, live it. We have heard it, and there is a very strong mental health component to it. I'm a little bit more, partial to mental health. I'm a neuropsychiatrist, so for me, I'm always thinking of, how does what happens to your body affects your mind, right? And how then it goes back and then, re-affects your body again. And it takes over, your whole persona, your whole life, your relationships and everything. So a lot of effects of wAIHA we have found, stemming all the way from the fears that are generating because of the symptoms returning, right? We have heard that quite a lot, and who better than you guys to know of that? Uncertainty, right? All the way, through different phases of the disease, from the diagnosis, from, your first visit to getting your treatment, and then, Sometimes the drug works, sometimes it's not working, it's the same drug. There are variabilities that are happening, so that's important. We've all discussed this, right? We all know of this. Unfortunately, to date, there are no approved treatments, either at the FDA or from EMEA for wAIHA. And there is a huge unmet need, right? There is a need to have approved therapies with proven safety profiles that can really help maintain the immune function of the patients, so they have better livelihood and better symptom control. In terms of the, we talk a lot about science. Dr. Panch presented this amazing presentation about the clinical phenotype characterization, the genomics of it, right? It took me back to grad school where I was when I was learning genetics. But, it's at one end you see that genotypic characterization, and at one end you see that phenotypic characterization, right? So the clinical piece is the phenotypic, the genetic piece is the science there. But the magic happens somewhere in between, right? Which is kind of the two levels coming together. And that is all about the experience. And the experience could be different for different patients. And it could be, differences when it comes to initial symptoms, differences that comes to treatment response, disease courses. There is a lot of variability in that in different patients. And how can we understand it better? The best way to understand it is from the patients directly, right? What is happening in your particular life? So keeping that background in mind, we really wanted to go ahead and create a patient engagement council, which we did. We're very proud of that. And we did a couple of very rigorous, qualitative research projects with them. And we're going to share some of these findings. These findings are coming from patients who are from the US, so there are findings from the US Council, as well as there is a global council. And as I shared earlier, some of these presentations have already been presented at ASH and at NORD. So I think I kind of covered it. It started from 2022, and then 2024, we're still doing some councils. So this is like, a continuous commitment of our organization to connect with the patients, to learn from the patients, to really help them, to help understand and then help, create innovative solutions for them. And it's all about understanding the lived experience of patients with wAIHA. We believe that this work is fundamental to our approach in terms of creating innovative treatments, because the more that we know in terms of the disease burden, the more that we know in terms of the treatment journey of individuals with wAIHA, and then their particular individual experiences that comes with their diagnostic odyssey, their treatment journey, their mental, emotional, and social impacts, would really help us to create a stronger understanding of the disease and how to facilitate treatments for those. Again, as I said before, right, it is a limitation, but again, right, when you say 1 to 300,000, they are going to be small quantitative samples. One thing to keep in mind, these are all self-reported diagnoses. A confirmed diagnosis of wAIHA could not be verified. There could be a recall bias, right? All of us, many patients have been having the disease for a number of years. So that's definitely, that could be there. These findings are coming from that select set of patients who were in the council. So generalizability, could be challenging to a wider population, but I'm sure, many of you, something will connect to you in your journey of what we see here. So let's take a peek first at the US Patient Council, and these were engagements that were done in 2023 and 2024. And this was presented at NORD. Dr. Piaczek was the PI for this work. So as you can see, a methods overview, right? Very scientifically rigorous. We had qualitative interviews with the patients. These were 2 hour virtual, patient council sessions that were done. there was an external organization who conducted these interviews. These were conducted first. So first was the collection of the patient, right? The recruitment of the cohort. These were from the U.S. with the self-reported diagnosis of wAIHA. They were screened, they were recruited, and when we started our project, we came up with an N of 6. So again, a small number. And then what we wanted to do, we really wanted to do 2 virtual focus groups with these patients. These were conducted by professional moderators with a discussion guide with all the rigorous checks and balances that are needed with the consents. And there were transcripts that were generated with the consent that work would be presented and published. We wanted to really start our work with understanding stressors, right? That were related to the experience of living with wAIHA. And then once the data was collected, we wanted to bring forward, our understanding of social epidemiology, the methods from social epidemiology, and how we could take that and then take the big chunk of data, right? And from those, can we glean insights, and would that help us to create thematics, which would then help us understand, what are the key stressors and themes that are identified from the observation. And those would be the insights, right? That would help us really understand wAIHA more. So with that methodology, firstly, understanding what the population is, as I said, 6 patients, and then predominantly females, right? 5 females and 1 male. We had more Caucasian patients, but we had 1 African American as well as 1 Asian patient. If you look at the age range, right, it's interesting. So we have quite a number of younger patients, right? And some which are a little bit older. So when we look at the literature and they say that you have patients more coming later on in life with regards to wAIHA, it's so interesting, right? And I think, Karen, you were showing some data as well, and Lisa and I, we were sitting and we were saying, hey, look at that. A third of the patients are women of childbearing age, right? It was so interesting, right? It was an aha moment. But when we go to the literature, it's all about, it shows in the 5th decade of your life or the 6th decade of your life, that's where you see. So we are learning and growing the literature together as we speak. There is a paper coming out right now, and I'm sure Dr. Irina would be working on a paper, I'm sure, at the moment, right? She's always creating something interesting, right? Very, very educated group, right? All, majority with bachelor degrees, with postgraduate degrees. So, we were very, very lucky to find very eloquent people, right? Who were very expressive, and, they had a lot of experiences that they could share. So more value to the work. And when we look at the raw transcripts, right, and then we analyze it, so there was 2 group of people. So we had a social epidemiologist who looked at the data, and then we had clinicians who would look at the data and then really decipher. So one is like looking at the data and trying to find how I kind of think like, when I put my data hat on, it's like looking at patterns, right? Like when you look at the totality of the data, looking at the big kind of scatter plots, right? Where is the majority of the data? And we find very clearly 4 kind of main buckets, right? There is uncontrolled symptoms and treatment AEs. I'm sure many of you would have experienced that. There is definitely a healthcare burden theme that emerges. There are daily life and aspirations, right, which are unmet, and definitely social support needs, right? These kind of connect all the time. When we look at the uncontrolled symptoms and treatment AEs, and again, you see the 3 points, so by no means does it mean that one is higher than the other, but this is how it's arranged. They were kind of even, right? We definitely see side effects and AEs with existing unapproved treatments. We're seeing instability and uncertainty, right? A lot of that. That was a constant theme. Whenever we would talk to the patients, they would always be talking about anxiety for the upcoming attack, right? They'd be like, we're fine now, but we don't know, right? When, we're good right now, thank God we're good, but we don't know when the next one is gonna come. Or one would say, I don't have it and I feel lucky that I don't have it, but my peer has it, and it's not a good state to be in. So the instability and uncertainty was definitely a strong theme. And then the illness brings this burden, this huge burden of the symptoms. Definitely, there was a lot of talk about accessibility to care, and I think, again, Karen showed some data earlier, right? We see a lot of patients in the East Coast, but maybe not so much in the Midwest, right? And we kind of connected, right, in terms of why that could be. There's not enough time in terms of education when they go and they connect with with clinical care. So that they feel education is definitely a need here. And then treatment, right? In terms of the existing treatments that are there is definitely burden in terms of accessing the treatment, in terms of the timing of the treatment. wAIHA has a huge impact on the daily life aspirations. It affects all areas of your life. It is your work, it is your family, it is your personal. So the whole 360 of your life is wAIHA, right? It affects every aspect of you. And it makes you very emotional. Why me? And what do I do? How do I get better? Are there more like me? Where do I get the support? There was a lot of talk about the need for information. And we've done some other research also. We did some social listening studies, which is not part of this, but just sharing, where we found a lot of questions being asked on the internet. Patients are asking questions to the clinicians, and then clinicians are asking questions amongst themselves, right? They're asking experts all the time. So these were the main themes that kind of came. So, very, very enlightening. And I think this is like a smaller kind of a piece. But some things, if you look at the selection of the quotes, right, it's hard to read everything. But what kind of emerged is these were some keywords that we pulled out, really hard, right? So the disease, the uncontrolled symptoms, really hard. Worst is fatigue. Those were the words that was back and back, right, back to back in the transcripts. They get angry a lot with their disease, with their state, with the care, with the treatments, and even with the support that they get. wAIHA kind of makes them extremely emotional, so they cry a lot. They feel that it's extremely draining, and we kind of touched on that, right, with regards to their work, with regards to their families, with regards to their relationships. There is always fear. It hurts. It's very depressing. There is a lot of ups and downs and relapses. It's really scary for the patients. And there are times when they feel extremely septic, as if they can't breathe. So, it's easier for me to come and read it, but you guys live it every day, right? And the patients that we talked to, they talked about it, and it was very moving. We definitely saw a huge mental health piece associated, and then the need for more and better social support needs, which could then help these patients cope up with their disease better. There is a huge unmet need in terms of effective treatments to help these patients control their symptoms, to have better AE profiles, so the risk-benefit, and then help them get better or more in control of their lives. If you look at fatigue, right, that came quite a lot. So as you can see, not every patient answers every question. Sometimes in some sessions, some patients don't come, right? So amongst the 4 patients who participated, as you can see, before disease onset versus after disease onset, the majority of them, they kind of feel that, there is a huge impact on fatigue for these patients. And when we looked at the data, we let the patients speak, right? We had some kind of high-level kind of ideas in terms of the discussion, but the discussion was free-flowing, and it was very interesting that when you talk to the patients, they think in their mind that there are 3 stages of their disease. after their diagnosis. Some of them feel that there is a period when they feel that they are in remission, definitely. And then there is a period which they call in cycle, which was the first time I heard it as a clinician, which is how they define it as kind of suboptimal level of therapy. And then one was active hemolysis, right? So if we see in remission, even in remission, right, there is still kind of a 20 to 30% impact on their life because of the fear, because of the anxiety. It's going to come back to me, and it drains them. When they have this suboptimal response, it can go as high as up to 60%. That okay, even drug is working to some capacity, but still there is a lot of burden. And then if you look at hemolysis across the board, it's like 80 to 100%. It takes over their life. So very moving data, which talks about the burden of disease, on the participant's life. When we talk about the impact on daily life and aspirations, it is very challenging once you find out that you have the disease, once it hits you, in terms of adjusting with the disease. It takes a long time for you to really cope with it and to adjust with it. There is a lot of missing work. And, it's challenging to how to modify your life, right? And how to get this special kind of accommodations for work. And some of them say that they have negative impacts at work. They felt that they were not treated fairly, but that's kind of their disease because there is lack of working accommodation. But other than the professional side, there is a toll on the personal side as well. What the patient was prior to wAIHA in terms of what their aspirations were, now it goes south, right? The aspirations are gone. Now it's like, how can I get the day going? How can I be maybe back to normal? Maybe 50% back to normal, maybe 30% back to normal. That's my definition. It takes a strain on relationships. We talked a bit with the patient caregivers as well, the care partners, right? And they also feel, so it's not, wAIHA doesn't just hit the patient, it hits the caregiver as well. And, there are definitely personal impacts as well. I don't know what's happening with my computer. Give me 1 second. All right. So overall, if we look at it, there is definitely a more need for social support because there is lack. That needs to be, a top priority. The mental health and the mental well-being has to be a part of the clinical treatment or clinical paradigm because there is anxiety and fear, which starts with anxiety and fear, but then there is comorbidity with depression and negative emotions as well. Psychotherapy or medical appointments or, when they go for that, those treatments, having a care partner with you has a better outcome because you have that support there. And patients feel that they really need to self-advocate for themselves to get access to therapy, right? Especially the patients who are in remote areas, like as we were discussing, right? 4 hours for a clinical visit, difficult. And with that, I will invite my partner, Lisa Shea, for the next part. Thank you so much.
Hi, everyone. For those that I haven't met yet, my name is Lisa Shea. I'm the Global Director for Patient Advocacy and Engagement at J&J, and I look forward to speaking to every one of you. So just really thrilled to be here, and thanks for everything that you do. So I have the pleasure of representing work done. A couple of authors actually sitting in the room with us. So Dr. Panch was a co-author on this abstract, as well as your very own Karen Jones. So thank you so much for your contributions here. I get to share insights from a global patient council. So I know I'm the last speaker between you and the reception, so I will just put an exclamation point on a couple of points that Zia had raised and we've been talking about throughout the day. So our Global Patient Council actually represented 8 participants. We skewed more towards male than female, actually, with the Global Council. And we had 6 US representatives, 1 from Hungary and 1 from the Netherlands. So as I go through my presentation, I'll highlight some of what sort of the global variances or regional variances were that we teased out as part of this research. Also diversity in race and ethnicity, and then we also looked at whether patients had previous clinical trial experience. Part of this Global Council, when it was created in 2022, was to help inform the development and protocol of our clinical trials. So understanding sort of where everybody was coming from and sort of their expertise in that area was just helpful to level set and kind of know where we needed to start as part of those discussions around clinical trials. The other piece that you don't see on here, but I think important to mention, is that we did have a variable disease course of the patients that participated. So patients were diagnosed as early as 1 year prior up to 21 years since the diagnosis. So a varied range of experiences, which I think helps to contribute to some really rich data for an N of 8, which we know for rare disease, as we've been talking about, is still really important. So the key areas of discussion across these 4 different council sessions that happened over the course of 2022 to 2024, we talked about initial symptoms, the road to diagnosis, efficacy and side effects of treatments, how the participants were managing their disease, the mental, emotional, and social impact that we heard about from Zia, and then also some of the experiences around relapse and remission cycles and thoughts there. So in terms of the results, we did see a variety of initial symptoms with our participants, from severe, acute, to slower and mild, and also a range of time to diagnosis, so from 4 months to 7 months to even 2 years. And I think for some in the room, it might be even longer than that as well. So really the range in diagnosis and the need for better diagnostic tools. And then 3 participants, important to note, as Zia talked about, is around the impact of fatigue. So they noted that there was a disconnect between their hemoglobins and how they actually felt physically. So really a call to action around not just focusing in on our numbers, but also how we feel and our quality of life, and assessing that as part of the treatment journey for wAIHA. The other pieces to call out, just in terms of global, not all treatment options were available across countries, so there was some regional variability around access to treatments that was interesting. And then from a US perspective, financial hardship also seemed to be a focus of some of the discussions as well. And then in terms of mental health topics, because we know, as we heard from Zia, this clearly comes out as important to all of you. We heard that peer support was really important, hence why we're all here this weekend as part of the wAIHA Warriors community, as well as hearing from the participants different ways that they dealt with mental health concerns, such as seeking counseling. Meditation was also mentioned, and then the advocacy community too was really important in those discussions. The 2 other pieces that we also teased out was this concern for relapse, as well as a fear that they would contract a virus like COVID-19 because of their immunocompromised status due to the treatments that they were on. So some important factors, I think, teased out there is sort of why this contributes to the mental health impact and anxiety that we see as part of individuals living with wAIHA. And then patient quotes, as Zia talked about, this was all qualitative research. So for us, really giving it the context to the data points, so to help understand what it's like to live with wAIHA on a day-to-day basis. So the first 2 across the screen that you see there is really around being unable to breathe, right? The shortness of breath, being terrified to exercise. Things that people often take for granted are a day-to-day common challenge that wAIHA individuals deal with. And then in the middle there, I think it really speaks to sort of the social productivity impacts of wAIHA. So This individual states that they had to stop working, right? And then that was also part of the financial hardship that they experienced. And then also the isolation. We had one patient talk about how they lost friends and family because they didn't understand what they were going through, and it felt like it was too hard to communicate what it was like to be living with wAIHA. And then the last piece, again, more resources and information is needed. This patient says more support would be helpful. Even the highly educated find this is a gap. And then some of the research materials, of course, this was a global council. A lot of the materials right now are in English, so it would be so valuable to have them translated, which I know Karen has worked on that effort, so really kudos to you for that. So just some of the key takeaways. I'm going to wrap up. So the first two are key takeaways from Zia's presentation from the U.S. Council specifically. Patients describing the stressors and themes related to uncontrolled symptoms, some of the adverse events, and then some of the medical care and access concerns that were teased out as part of that discussion. Social support and how critical that is to help alleviate these stressors, to help improve their overall mental health and quality of life. And then the last piece was a finding that we really highlighted in the Global Patient Council work around the increased focus by providers that's needed for them to focus on mental health, providing services, critical connection points to those living with wAIHA, which of course also includes resources from patient advocacy like the wAIHA warriors themselves. So again, just really grateful to be here for this with this community and partnership. We hope this presentation kind of helps you understand that we hear you. We're very grateful for your willingness to offer your time and expertise so that we can elevate your voice and really drive the need for new innovations and enhanced care in this space. So thank you again. And we're happy to take questions during the reception that's next. So thank you so much.
So I'm very excited to announce Dr. Irina. She's an associate professor of hematology. She completed a medical degree at Albert Einstein College of Medicine and residency and fellowship training at Montefiore Health System. Her clinical and research interests focus on autoimmune hematologic disorders, including autoimmune hemolytic anemia, paroxysmal nocturnal hemoglobinuria, and immune thrombocytopenic purpura, as well as hematologic issues in pregnancy. Dr. Irina is currently a chief of the Division of Hematology at Montefiore Medical Center.
Thank you for this very kind introduction. So today I wanted to talk a little bit about what triggers autoimmune hemolytic anemia. Not that we know what triggers it, but we can try to hypothesize, and I will share with you guys at least a few things that we know about it. So we know that warm autoimmune hemolytic anemia is more common in adults. The median age is 52 years old, but the incidence is increasing with age, just like other autoimmune disorders. And we know that part of it is actually aging, and aging cells are more immunogenic, so they're more likely to trigger the immune system. And women are more commonly affected, again, for whatever reason. Majority of the diseases of the immune system tend to preferentially affect women. And I don't want to belabor it, but you guys probably already know that the main pathophysiology in warm autoimmune hemolytic anemia is production of pathogenic antibodies by the B cells. Most of these antibodies are of IgG subtype. They bind to red cells at the body temperature. That's why it's called warm as opposed to cold, which is triggered by antibodies that bind to red cells at the temperature below core body temperature. And then sometimes these antibodies can fix complement. That happens about 50% of the time. And that leads to red cell destruction. So these antibodies are produced by the types of cells called lymphocytes, which are main part of the immune system, as well as plasma cells under control and guidance of T cells. And we'll talk about what all of these things are. So this is a complicated slide that basically describes the pathophysiology of warm autoimmune hemolytic anemia. So as you guys know, you have your red cells covered by antigen that for whatever reason the immune system doesn't like. It's really the same antigen in most people. It's called band 3. The antibody binds to this antigen, and then it goes usually in the spleen. Spleen is kind of a cemetery for red cells, which is why a lot of times splenectomy works in warm autoimmune hemolytic anemia. And in the spleen, you have this phagocytosis. You have this interaction of antibody-coated red cells with the Fc gamma receptor, so that's like a special receptor on the surface of cells that like to eat red cells, they're called macrophages. And then the macrophages eats the red cell, and that's how it happens. So what causes this? What are the causes of immune problem that leads to autoimmune hemolytic anemia? So, of course, as usual, everything in medicine, it's multifactorial, right? So there is a lot of things that cause immune dysregulation. And it starts with a predisposition to an immune dysregulation, potentially genetic. And again, although most of the time, about 50% of the time, we don't know what causes autoimmune hemolytic anemia, we do know that certain immune deficiencies and certain mutations that have to do with the genes in the immune system are associated with higher risk of autoimmune hemolytic anemia. We also know that a lot of times autoimmune hemolytic anemia or ITP or other autoimmune diseases flare or happen for the first time after a viral infection, right? So the viruses and things like that can cause hemolytic anemia. And then there are other things like neoplasms, other autoimmune disorders, as well as medications. And again, we'll go through the details a little bit more. So that leads to, the fancy term is breakdown of central and peripheral tolerance and destruction of red cells. So really basic about the immune system, right? So immune system consists of two main parts, the innate immunity and adaptive immunity. Innate immunity is kind of non-specific. It's your neutrophils, it's your basophils, what else is there? Dendritic cells and macrophages, eosinophils, a bunch of white cells that are responsible for killing macrophages, but they're responsible for killing microorganisms. And each one of them actually has its specificity. So like, for example, eosinophils like to eat parasites. And neutrophils like to eat bacteria, right? So each one of them are specialized, but that's all the specialization they have. They can't recognize one bacteria from another bacteria, right? They just know it's bacteria and they gotta eat it. And then there is adaptive immunity, and that's the immunity that can adapt to a particular antigen. So it can recognize one virus from another, self from non-self, and adapt to that. And that immunity is evolving as your body matures, grows, as well as gets old. And the organs that are involved in the immune system are basically the lymph glands, right? The bone marrow, and the thymus. Those are the main organs that have to do with the immune system. So autoimmune hemolytic anemia, just like the rest of the autoimmune disorders, results from breakdown of this central as well as peripheral immune tolerance. So what am I talking about? What is tolerance and why does it break down? So central tolerance occurs in the thymus and in the bone marrow. That's where the cells start developing. Thymus, T cells, bone marrow, B cells. So all the cells initially start, all the blood cells are made in the bone marrow, and then they go to their respective places for further growth and development. So then they go to college. So for T cells, college is thymus. And like I said, for B cells are bone marrow and lymph nodes. Peripheral tolerance occurs in peripheral tissue, so lymph nodes, where the T cells that already, went through kind of their school of central tolerance, they basically police other self-reactive cells and make sure that they don't cause trouble. And that's the peripheral tolerance. So the key players are all kinds of lymphocytes, right? Those are the adaptive immune cells. And the main lymphocytes we're going to talk about are the cytotoxic T cells, B cells, regulatory T cells, and CD4 helper T cells. And the name is kind of tell you what they do, right? So helper cells help the immune cells, cytotoxic T cells kill all the other cells, and then B cells produce antibodies. Regulatory cells regulate things. So again, central tolerance, I know I'm repeating myself, but this way we go slow and we go through this, and you guys will know all of the immunology by the end, right? Okay, so a naive young T cells goes to thymus, college, right, and starts maturing and developing. And the first thing that happens, it acquires a receptor. It needs a T cell receptor. Whatever it needs to do, it needs to interact with things. So it acquires a T cell receptor. And the cell that acquires T cell receptor gets kept. If you don't acquire T cell receptor, we don't need you. It gets cut, so it gets deleted. That's called positive selection. You positively select the cells that have the T cell receptor that interacts with molecules moderately well. Not too good, not too bad. If you don't interact with molecules at all, then you don't get selected, right? Like if you didn't find your true molecule that you're supposed to kill, right? So the T cells basically go to school to learn to kill other cells. So if you don't find the molecule that you're gonna kill, we don't need you, then that T cell gets neutered. If you find if you have too much of an interaction with those molecules, so let's say it's a self cell, right? Your T cell in the thymus is looking at a piece of a cell, a piece of your own tissue, basically, and it's a strong affinity. That cell gets deleted as well. So basically, in the thymus, the main two things that happen, you select the cells that are able to have a functional receptor that interact with moderate affinity with MHC molecules. And I'll tell you a little bit more about what the MHC molecules are. If they interact too strongly, they get deleted because you don't want to self-react, right? And if they interact not strongly enough, they get deleted as well. This is a more complex view of the same thing, right? And the reason I wanted to show this is because there are a couple of molecules that you guys will see and the names that you'll recognize, but you definitely don't need to remember it. So again, in the thymus, you have these interactions and you select the cells that are able to react and you kill off the cells that don't react. So, and then that react too well. And then the T cells come out of the thymus and they have a couple of different paths. So the ones that react against their own tissues die. The ones that survive and react in a moderate way become regulatory T cells, so they become these policemen. And then there are a few T cells that really escape that still are able to react against self-tissues, but they just kind of sneak out of the thymus and end up in the periphery. And in the periphery, these T cells basically, periphery is other lymph nodes and a bloodstream. These T cells develop a little bit more and acquire some other functions, and they become effector cells. So those are the killer cells, right? And they become helper cells. So those are the cells that are gonna help the B cells. And there's a lot of molecules involved here. So one of them is FOXP3. and that is mutated in one of the autoimmune disorders called IPEX syndrome. And there are CTLA-4 molecules that are important as well, and we'll talk about them as well. So, like, these molecules are important. So, same thing happens with the B cells. So the B cells initially go to the bone marrow, and if they recognize their own self-antigens with a very high affinity, they get deleted, right? You don't want your B cells to react against you. If they don't react to the B cell, to the self-antigens, they get deleted as well. But if they have a little bit of reaction, if they're able to recognize self-antigens but not attack them, they get kept. And then same thing happens in the periphery, right? So the B cells go in the periphery and they further learn how to recognize self from non-self and not to attack self. And that not attacking self is called tolerance. So what does actually happen when you get a bug? Why are we talking about that? So there are cells which are called the antigen presentation cells, which when they encounter an antigen, and that antigen could be a virus, a bacteria, or a red cell, they eat it, and then they kind of regurgitate it, right? And they present it on their surface in a molecule that's called MHC molecule. Remember, those were the MHC molecules in the thymus that were reacting with the T cell receptor. So they present it to the T cell and say, hey, T cell, look what I ate. And T cell looks at it and it's like, oh no, that's not good. We need to get rid of this. This is a dangerous bacteria, right? So the T cell recruits other cells, including helper T cells, as well as B cells, and they proliferate. There is more of them, and then they secrete little cytokines and kill off the bad cell, and the cell is dead. Same thing happens with the B cells, except the B cells make the antibodies that make the pathogen attractive. The antibodies code the antigen, and it becomes very attractive to the immune cells. The other thing T cells do is they activate B cells, so T cell and B cells, right, to produce plasma cells and memory cells. And the reason we need to do that is we need to remember those bacteria and those antigens. So remember that bacteria that caused the really bad flu 5 years ago? You still want to remember that, right? So there is an interaction between, again, don't think too much about all this mumbo jumbo, but there is an interaction between antigen as well as B cells and the helper T cells, right? That's the helper T cell. And you need all of these interactions for the process to go smoothly. If this CD40 ligand, which is called co-stimulatory ligand, doesn't exist, that interaction doesn't go properly. So either you stop reacting to the cell or you kill off the cell, depending on other things. So all of these factors need to be in play. And these factors, those co-stimulatory molecules, are called immune checkpoints. This is where your immune system has checks and balances to make sure that you don't attack your own immune cells. So this is a regulatory mechanism that helps maintain self-tolerance and prevents excessive immune reaction, because all of the autoimmune disorders are effectively excessive reaction to self. And the main immune checkpoints are those CTLA-4, which basically are responsible for making sure you have central tolerance. So they're expressed in thymus, and they compete with, if you go, let's see, okay. Anyway, there is they compete with the receptor on the B cells, on the T cells, I'm sorry, over here. So if you pick, sorry, you have CTLA binding, that would be an inhibitory signal, and it inhibits T cell activation. But if you don't have CTLA binding, then you bind to CD28, and then it's a positive interaction. Same mechanism, but in the periphery, is mediated by PD-1 and PD-L1 interaction. Again, it's another co-stimulatory molecule, and it's the picture here has a cancer cell because we use these molecules right now in fighting cancer cells, PD-1 and PD-L1. Basically, those are brakes on the immune system, so to fight cancer cells, what we do is we take off the brakes and we allow the immune system to fight the cancer. But for the autoimmune diseases, it's the opposite, right? You want to have the PD-1 and the PD-L1 because they prevent excessive reaction to our own cells. So bottom line is there is a lot of moving parts. There are those T cells, there are B cells, co-stimulatory molecules, the molecules that make you more reactive, the molecules that make you less reactive. And there are lots of errors, and we're still learning most of these errors at each point of this immune tolerance that results in the development of the immune disease. And it's called loss of the immune tolerance. So the central immune tolerance failure happens in the bone marrow or in the thymus, where self-reactive T or B cells escape the deletion. And as I told you, a little bit of that is actually normal. And then the peripheral tolerance breakdown is when regulatory T cells actually don't do what they're supposed to do, right? So for whatever reason. Those are the reasons why that can happen. So again, genetic predisposition. There are germinal mutations that can impair immune system function, including congenital immune deficiencies. There are defects in FAS-mediated apoptosis, and that's a syndrome called autoimmune lymphoproliferative syndrome, which is associated with a lot of autoimmune disorders. There are defects of T and B cell interaction that cause autoimmunity. And as I've mentioned, these co-stimulatory molecules, CTLA4 and PD-1, also involved in immune checkpoint, and defects in those can result in immune disorders as well. People who have regulatory T cell dysfunction, for whatever reason, develop autoimmune hemolytic anemia as well, and we can actually see that in patients with warm autoimmune hemolytic anemia, there is not enough regulatory T cells. So we traditionally have been thinking that we make too many antibodies, and that's the main problem for autoimmune hemolytic anemia, because the antibodies destroy cells. Well, maybe the problem is that we are not controlling our B cells, and that's why they make too many antibodies, and it's really a T cell disease. Our immune system gets confused. So if you have a virus, and that virus looks like your own tissue, it gets confused, and it can, by mistake, attack your own self-tissue because it thinks it's a virus. That's called molecular mimicry, and that's another reason why people develop autoimmune diseases. You found, you picked up a virus that just looks too much like one of your cells. Polyclonal activation, so conditions like lupus and other inflammatory conditions. Your immune system is just irritated, and it's just making antibodies and making antibodies and making antibodies, and certain things just get in the way. So that's another mechanism of warm autoimmune hemolytic anemia. And then finally, cytokines. Cytokines are little things that are floating in the blood that signal cells to do different things. They're like little soluble signals that float in the body. And we already know that certain cytokines, certain syndromes are associated with warm autoimmune hemolytic anemia. And this cytokine imbalance, either because of the genetic reasons or acquired reasons, can cause hemolysis as well. So basically, again, nothing that you need to necessarily remember here, but to give you an idea of this is the process, those are the mutations, and all of these are genetic mutations that can be associated with autoimmune hemolytic anemia. That being said, all of this is very rare. So those are the pathways that we think are important, but this is highly rare. Now, why some people are more predisposed to autoimmune disorders than others? If you start asking, right, everybody has a member who had rheumatoid arthritis or multiple sclerosis or lupus, or there is a cousin who has one of those things. So another reason why people are predisposed to autoimmune diseases is they have genetic variability in antigen presentation. So your T cell will not recognize an antigen by itself. It needs it to be presented. It wants it on a plate. That plate is MHC, major histocompatibility complex. And basically, your immune cells only recognize antigens when they're presented in a context of this MHC molecule. And people differ in the way these MHC molecules present the cells. Some MHC molecules are more effective in presenting cells, and this is a genetic variation as well, right? So some MHC molecules are so effective in presenting cells that they can cause autoimmunity. Fast ligand is another mechanism where you get rid of autoreactive cells, and it happens very early on. And there is a condition, as I've mentioned, autoimmune lymphoproliferative syndrome, where you have mutations in this ligand, and you do not get rid of self-reactive cells. And that's associated with a bunch of autoimmune disorders. I kind of mentioned the polyclonal B cell stimulation when you have activation of B cells by lots of different stimuli, which could be, for example, infectious agents, HIV or COVID, or Epstein-Barr. And that just gets the immune system going. Molecular mimicry we've discussed as well, right? The self-antigens just, yes? 5 minutes, okay. The self-antigens look very much like the bacterial antigens, and you get confused, and you attack your own cells. The emergence of forbidden clones. So in certain cancers, right, lymphomas, they produce clones of abnormal antibodies. That's usually not the case in warm autoimmune hemolytic anemia. It's a polyclonal disease, meaning all these antibodies come from different parents. Unlike cold agglutinin disease, where all the antibodies come from one parent. But in warm autoimmune hemolytic anemia, they're all polyclonal. However, in some situations, you have lymphoproliferative disorders that make these clonal cells that escape the normal immune tolerance, and because their antigenicity is a little bit altered, the protein is a little bit different than normal, we don't recognize them as foreign, and we keep it. You can also have really strange epitopes, really strange antigens that immune system just doesn't know how to recognize, and create self-reactive cells. So what is, again, the importance of this in real life, right? So we just talked about you can have errors early in development, errors in late development. How is it important in real life? So if you look at, there was a study that looked at 47 patients 47 adult patients with immune thrombocytopenia, autoimmune hemolytic anemia, and autoimmune neutropenia. And they were all young at the onset of immune cytopenias. And they were able to find these lots of different mutations. The fancy term is inborn errors of immunity in 19 of 47 patients. So less than half. And again, realize those were patients who were diagnosed at a young age. So the genetic issues are more common in people who are diagnosed at a young age, right? I mean, if the disease happened when somebody was 70 years old, it's just less likely that there was a genetic predisposition to that. It would have revealed itself sooner. So out of those 47 patients, the most common autoimmune conditions were common variable immune deficiency, the ALPS, autoimmune lymphoproliferative syndrome that I've mentioned before, and then a few other ones. And they were associated with low numbers of T and B cells as well as other immune abnormalities. I want to mention Evans syndrome, which consists of two autoimmune cytopenias. It's usually warm autoimmune hemolytic anemia and ITP. In children, 65% of patients have genetic abnormalities. In adults, more like 11%. So definitely not as much. Or maybe we just don't know. My belief is these abnormalities are just more subtle and we're just not picking them up yet. If you look at the family, at the associated autoimmune disorder among these people, they have 14% of associated immune disorders in the first degree relatives. And actually, I don't think I put it here, but when we did our study in patients with autoimmune cytopenias, family history of autoimmune disease in the first degree relatives was about 15%, which I think is a little bit underrepresented. So the clues that somebody has one of those inborn errors of immunity are at young age at diagnosis, if somebody has difficult relapsing refractory course, Evans syndrome, obviously, as we mentioned, and lots of abnormalities on the immunologic tests and multiple autoimmune disorders. The diagnosis that, the way we make the diagnosis is next generation sequencing. We basically check the DNA. It's a DNA sequencing. I want to say a few words about common variable immune deficiency, since it is the most commonly associated immune deficiency with warm autoimmune hemolytic anemia. You basically don't make B cells properly, and that results in immune dysregulation. So your immune system doesn't work properly, and when your immune system doesn't work properly, it doesn't do its job. So it doesn't, you don't have the normal surveillance for cancers as well as infections. But at the same time, because it doesn't work properly, and it's because of those regulatory T cells, right, that don't police other cells, you also get a lot of autoimmune manifestations. And although this is a congenital disorder, majority of people are diagnosed in adulthood, so this is not a disease that is seen by a pediatrician. And actually, majority of the patients are missed. I have patients that are diagnosed at 60 years old, 70 years old. If they don't have the typical presentations, which are recurrent infections, usually sinus infections, it's easy to miss. And a lot of patients with common variable immune deficiency have lots of different autoimmune disorders, and like I said, lymphoproliferative disorders as well. Diagnosis is simple. The gamma globulins are low, so IgG is low, IgA is low, sometimes IgM is low. Autoimmune lymphoproliferative syndrome, I've mentioned a few times. This T cell development involves induction of apoptosis, which apoptosis means cell death, by this fast ligand, which is deficient in patients with autoimmune lymphoproliferative disorder. It's fairly common, and it results in, so you don't kill autoreactive cells, so it results in their accumulation, and including B cells, they produce antibodies and cause, again, lots of problems with immune system. This is just a mention of some other inborn errors of immunity, which are nearly not as common, but just something to know that a lot of inborn errors of immunity also associated with endocrine dysfunctions. So just to mention that. The cytokines, which are little soluble syndromes that are going between the different cells to communicate. Their abnormalities in production also are associated with immune dysregulation, and of particular relevance, I guess, to this group. So you guys know about the rituximab, right? So a lot of times we use rituximab for treatment of warm autoimmune hemolytic anemia. Well, when we use the rituximab, it basically, B cells need to, there's a cytokine called BAFF, BAFF, and B cells need this BAFF to survive. They need to eat BAFF to survive. But there's not enough BAFF in the system under normal circumstances. And that kind of restricts the B cells. But then when you give patients Rituxan, kills off all the B cells. So now there is no more B cells to eat the BAFF. So 1 or 2 B cells that survive the rituximab starts eating this BAFF and and proliferates. And if that's the B cell that was producing antibodies that was killing the red cells, that's how your immune system escapes. And that's why the BAFF inhibitors, for example, ianalumab, are currently being studied in warm autoimmune hemolytic anemia, because that's one of the ways the immune system kind of fools us after the rituximab. Yes, this is, as I've mentioned, the study that we did looking at association of autoimmune disease and warm autoimmune hemolytic anemia, as well as other autoimmune disorders. And we saw history of another autoimmune disease in 12% of patients with autoimmune hemolytic anemia, and family history of autoimmune disease in 16% of patients. And most common was lupus, and Sjogren's and rheumatoid were close second ones as well. We already have some treatments for errors of inborn errors of immunity. So tocilizumab, so that's one of the anti-cytokines, PIK3 kinase inhibitors. Mycophenolate may work for a subgroup of people, so we can actually, even for people who don't have inborn errors of metabolism, we can kind of subdivide people depending on which pathway is active. So we do these fancy tests, checking lymphocytes, checking cytokines, and that gives us an idea of which one of the pathways is active, and we can actually try to come up with a targeted therapy. Like, for example, patients with ALPS that I've mentioned respond well to sirolimus. And it is like looking for light in the dark, but we're almost there, and I think soon we'll have at least a better treatment, if not the cure. And that's all I have for you guys.
So next I have the pleasure of introducing Jen Raber, and just reading her biography, I learned a lot that I did not know. So Jen was born in Chicago, but spent most of her formative years in Northern Virginia. After graduating with a Bachelor of Science in Psychology from James Madison University, she went on and obtained her Master's in Forensic Psychology from Castleton State College in Vermont. Jen has made Cincinnati, Ohio her home for the last 23 years. Since the FBI was not ready for another Jodie Foster-esque agent, Jen decided to move into banking and payments in the fraud and anti-money laundering space. Jen currently works for Protiviti, a global consulting firm, as the lead for the Financial Crimes Compliance Department at the Americas Delivery Center. She went back to school and graduated in December of 2023 with her MBA from Northern Kentucky University. She's been married to Scott for 6 years and has a daughter, Abby, who graduates from high school next month. Abby's going to begin Abby, being extremely creative, has decided to attend Paul Mitchell Academy in cosmetology. Jen, Scott, and Abby also share their home with the three best girls in the world: Sam, a 9-year-old German Shepherd; Zia, a 6-year-old boxer mix; and Stitch, a 1-year-old French bulldog. In her spare time, Jen has turned her love of traveling into a side hustle as a travel agent. So Jen is going to lead us through a panel discussion on the hope of remission. So please welcome Jen to the stage.
Good morning, everyone. My name is Jen Raber, and I am a patient living with warm autoimmune hemolytic anemia. When we first proposed this panel, it was because we, at some point in our journey, whether that's now or in the recent past, have been in remission, okay? Let's talk about what it means to be in remission. So first you have full remission, which by definition means you're symptom-free, you're not hemolyzing, and you're not on any treatment. And we have people in this room who are in full remission. Then we have partial remission, which to me is not much different. It means you're symptom-free, you're feeling good, but maybe you're on treatment. Maybe you've had rituximab, maybe you're on a medication, but you are symptom-free and you are living your life almost, if not exactly, prior to being diagnosed with wAIHA or CAD. So I am joined by 4 amazing women who we'd like to talk a little bit about remission and what the experiences have been like. And this is to provide hope. So whether you're in remission right now or not, there absolutely is hope for the future. So I would like each of these wonderful women to just introduce yourself and how long have you had wAIHA or CAD? And what kind of remission would you say you're in? And let's start with Janelle.
Am I on? Okay, my name is Janelle Glidewell, and I serve as a patient advisory board member. I've been diagnosed for 3.5 years, and thanks to Dr. Irina, I've discovered today I have aged cells. I'm so excited. That's basically it.
Janelle, would you say, what kind of remission are you in? Are you in remission right now?
I am in remission with the use of CellCept.
Great. Joanne?
Hi, my name's Joanne. I'm in partial remission as of 7 weeks ago, and I live in Pittsburgh half the time in the winter near Orlando, and I have cold agglutinin disease.
Thank you. Kathy?
Hi, I'm Kathy. I was diagnosed in '99 with wAIHA. I am in, compensated remission? I am still hemolyzing, however, my hemoglobin levels are normal, and I do take medication every day. My hematologist says, unfortunately, I am probably going to be hemolyzing for the rest of my life. So, that's it.
Great. Joe?
Hi, my name's Joe Banta. I have cold agglutinin disease. I've had it officially for 5 years, but we know now I've had the symptoms for decades. I live in Syracuse, New York, which is where you do not want to live if you have CAD. And I think I answered everything for the first question. What else?
Do you consider yourself in remission?
Oh. So it's hard for me to say remission because my hematologist doesn't believe that people with CAD are in remission, but I really feel like I'm in partial remission. Finally, near the end of 2024, all of my blood work was normal. Thank you. I had rituximab in February of 2022, so it's been a long journey.
Thank you. So for those of you, feeling in remission, and again, to me, that's symptom-free, right? Like no more fatigue or less fatigue, less headaches, less dizziness, less short of breath. How long would you say it took you to achieve your most recent remission? Kathy?
Okay, I had a splenectomy in 2011. I dipped to 9, my hemoglobin, in 2012, so I'd say 23 years. 13 years. I'm sorry, in the math. 13 years I've been in quote-unquote remission. My doctor says remission. I say that it's dormant. That's just my terminology, but that's how
But you're feeling good.
Yeah,
For
feeling
13
good.
years.
Yes.
It's amazing.
Yes.
Joe?
I started feeling much better right after I had my infusions, but my progress was extremely slow. I was in the low 9s, and it took about 4 months to get up to 10. And then at 11 months, I had a hemoglobin of 11. And I just, I think you get used to being at that low level, and then every time it increased, it was kind of like, wow, I can change the sheets on the bed by myself. So with every increase, I just felt better and better.
That's great. Janelle?
I would say it took about 18 months. I was initially, my story is very unlike other folks. I was called immediately to my hematology office. He said, "Do you know what you have?" And I said, I had done a lot of Googling with my lab results, so I had a good idea it was wAIHA, but I thought it's no big deal. I'll take a pill and I'll feel better. Well, no, that's not exactly what wAIHA is, as we all understand it now. 4 days later, I started on my first 4 rounds of rituximab. 5 months later, while on prednisone, 5 months later, they started a second round, both which did not work. So that is no longer in my future. They transitioned me to CellCept, 2,000 milligrams a day. And I will say that at about the 18-month mark of my diagnosis is when I started feeling better, still on low-dose prednisone, but I started feeling better, thinking there is hope. Because up to that point, when you have never been sick and you're just sick all the time, it's discouraging. It's depressing. But about 18 months ago is when I really felt I turned around and feel really, really good.
That's great. Joanne?
This morning, I wrote some notes after I saw your questions. So my case is a little bit complicated, but I just achieved the partial remission only 7 weeks ago, and I feel wonderful. And I've had every type of chemo and immunotherapy known to CAD for the past 3 years. I've had plasmapheresis, and I get that every 3 weeks. With CAD, you have the symptoms that's complement-based, and with the B-cell symptoms. So for years, I've taken Enjaymo, and I get that infusion every 2 to 3 weeks. And that had made a big difference in my life. The short of breath, the fatigue, my hemoglobin rose, and just like Joe, you're just so excited, to see that. You just can't believe it. And just lately, my hemolysis markers have normalized. So they're completely normal. So with that, and with Sean's help, he directed me to a hematologist at Moffitt Cancer Center. And she started me on daratumumab that Dr. Panch talked about yesterday. And for years, I've told there's nothing else we can do for you other than the Enjaymo, but the B-cell symptoms had to be mechanically removed with the plasmapheresis. So after 6 years, I just can't believe it. I just can't believe it. I haven't had plasmapheresis now for 8 weeks. And, I told them I was at Washington, D.C. Last weekend, and I trotted around Washington for 2 hours, sightseeing, and I kept telling my husband, look, I'm not short of breath. Look, I don't have any symptoms. And we kept going on and on and on and on. And it's just amazing that I feel, that I'm like almost normal, but I don't mind the NGMO infusions. Like I said, that takes care of that part. But with the daratumumab, it's a real game changer,
That's amazing.
I know.
I'm so happy for you.
I know.
For me, it's been an up and down journey. Diagnosed in 2020, typical, started on the prednisone, had the Rituxan, and I would achieve periods of what I would consider partial remission. 10 months after my first round of Rituxan, I was fine for 10 months. Then I would crash again. They tried another round of Rituxan, that didn't work. They tried cyclosporine, that didn't work. Well, it had a lot of side effects. I had my spleen removed. 2 months later, I relapsed due to what I personally believe, and my hematologist tends to agree, is a COVID vaccine booster shot. After that, I was also placed on Cellceptin, and I'm pleased to report my hemoglobin's been at 13 for the last year. So now I'd like to discuss your symptoms and your activity level after achieving some sort of remission. So Kathy, can you tell us a little bit about how you are now, in terms of your symptoms and activity level?
Symptoms, I pretty much have none. There are times when, if it's been a long day, yes, I get tired, but I think that's just part of normal life. Activity level, since I had my knee replacement back in October, my activity level is just shot through the roof. I mean, I could walk, and I'm ready. Where's Jim? I'm ready for DC again, and I could walk around. I'm like a little kid in a candy store, just running around, what I couldn't do before. And when your levels are up, you just have that burst of energy. And unfortunately, on another note, when they're going down, you could feel that energy slowly going, and something's going on. But it feels real good to be in remission and have normal levels.
Awesome. Joe?
So I still do have symptoms, even though everything's normal. I still have a cold agglutinin titer. And what's really kind of odd is it had really dropped after my treatments, but kind of the blood work and the titer haven't gone along the same. So I still was having abnormal blood results and high retic count for quite a while. And so now my blood results are good, but my titer is starting to creep back up. And so I do practice extreme cold avoidance. And this was a bad winter in Syracuse. And so there was actually kind of a month where I did not go out of my house. I used Instacart and things like that. And I had the biggest increase. My hematologist during winter tests me every month. He gets worried about all of his CAD patients in winter. And I had the largest jump in hemoglobin during that month. So I still have to avoid the cold and I have symptoms, when I get hit with cold. I have what I call a hemolysis event, and I have severe fatigue. But since this time, with the higher levels of, or the normal levels, and the higher level of hemoglobin, I get better much faster, and I feel a little more confident. I'm very social, and when I get sick, of course I have hemolysis. So I've been able to go to book club more, do some social activities without that fear. I used to get panicky the minute I'd feel cold on my face. You know, I'm getting over that. So, so that's, and I'm able to do almost normal things. I love to garden. I'm able to trim the bushes, and I couldn't do any of that before.
Sounds like you need to move to a warmer climate.
Air conditioning. Air conditioning's a problem.
Isn't it good to hear someone
Yes.
say, when I can do something else, I feel so much better, and that fear doesn't overtake you? Remember those feelings of fear when you were first diagnosed? Before my diagnosis, our adult daughters had difficulty keeping up with my pace. It changed very quickly. I wouldn't say that I'm back to that point yet where they couldn't keep up with me, but I would say that probably 90% of my life prior to my diagnosis is where I would be today. But I have learned something really important, and that is to listen to my body. And if my body says, you can't mow both the front and the backyard, or you can't, well, I don't mow, my dear husband does that. Sometimes under protest, but he does mow the yard. But just to be able to go to sleep at night and say, man, it's been a good day. I maybe didn't get everything on my list done, but I got some of it done, and I'm not wiped out. Or wake up in the morning and say, it's going to be a good day, I'll accomplish what I can. But overall, I am thrilled to be where I'm at right now. I attended my first conference not long after my diagnosis, and I thought I looked pretty decent. Well, I really didn't look decent at all, did I, Karen? And she was so sweet. She didn't ever say anything. And I'm just trying to keep up with the pace. And finally, on Saturday night, I said, Karen, I really need to go rest. And she understood it and was very gracious. And then I get this email. If I didn't tell you, you looked 100,000 times better than you did last year after the second conference. And I thought, Jay, did I really look that bad? You know, I thought I was beautiful, but apparently I showed the initiation to the journey of wAIHA. And I'm so happy today to be back feeling even better and stronger, and so thankful for Karen and her honesty with me.
Thank you.
After listening to the other ladies, you know, I think about it too. 6 years ago, I couldn't even walk down my driveway and get my mail. I was so short of breath. And you know, Karen, I've been up here the first one. You were saying think of positive things, and it was like doom and gloom for me. And here I am, like 7 weeks. I can't believe it. I can't believe it. And thank you, Sean, for helping me. But that's why I told you before I was in Washington, D.C. I mean, I'm totally normal. Like in JMO helped me along the way. With the shortness of breath and the fatigue is just unreal. You really don't talk about it, you know, when the doctor asks you about your symptoms, because it's always there. And then with the DARA, it took away, just like my plasmapheresis symptoms, and you know, you get short of breath and dizzy and night sweats and all that, you know, is gone. And then when you asked me to be here, I was like, I can really talk about remission. I would never think that of, you know, 6 years ago. So it's a really life changer. So sitting here from the first to the third one, I can't believe it. So I'm looking forward to this summer. We have 3 acres, and you know, even trying to walk in the yard, I couldn't do that. I was all short of breath. So I'm looking forward to some yard work.
Thank you. For me, and I think Sean captured it perfectly in my bio, that I'm doing everything. Before my diagnosis, I was a person that did a lot. I was a person that was active with church and very social, and I worked a lot of hours, long hours. And when I was first diagnosed, and the times I have not been in remission, it all stopped. Seeing my friends stopped. Working stopped for a while. And I couldn't do the things I wanted to do. I'm a person that I love to fill my life up with stuff, and my husband hates that. But for the last year to year and a half, I'm back. I'm back at going to church and being active. I'm back at seeing my friends. I'm doing the side hustle. I am working the hours and actually go to the office 3 to 4 days a week. So, my activity level, while not exactly back to pre-diagnosis levels, is very, very close. So much so that I'm starting to work out again. Before I was diagnosed, I had a Peloton, right? Like COVID, Peloton. Once I got diagnosed, we sold it. I was so short of breath, I couldn't bike, I couldn't exercise, I couldn't do anything. And now I'm back to, slowly starting to work out. So, it is possible. Last question I kind of want to talk about is, would you say your path to remission has been linear? And I could probably guess the answers, but if it has or has not, why? So Janelle?
I feel like and I kind of alluded to this, my pathway was linear for the first 18 months when the medicine wasn't working. And, I really didn't want to give anybody my spleen because I don't like pain. And so but after 18 months, it has just, on a graph, I would say it has just had to go like this. There's been no setbacks as of yet. Every day I seem to be getting stronger. And along with that, every day my mental attitude is improving because it seems the good days are so much more consistent than those that were full of questions.
Thank you. Joanne?
When I was writing out this question, I told my husband, I said, linear. I said, it looks more like the stock market. It's just, that's how I felt my life was, not linear. And no, it has not been linear for the past 6 years. I think for all CAD patients, the treatment varies significantly as the doctor spoke. And for the past 2 years, I had success with Enjaymo. And Enjaymo's only been FDA approved since February 2022. So like I said, the Dara's just been added to my regimen and, the success I've had. So I think staying strong and hopeful and, during any treatment is a journey that requires resilience and focus on physical, mental well-being. So, you have to seek out support, healthy lifestyles, and this is the best support ever. And I remember telling Joe, I called you after the first one. I said, you have to come. You have to come. I said, you're in a room with the people that experience the same thing. And it's just amazing, amazing. So, it's very helpful to keep that in mind, during all your challenging times. So there is hope.
Okay. Janelle? For if your path has been linear?
Yeah.
Oh, yeah. I'm in,
It was
Kathy.
like this.
Oh, sorry. Kathy.
Excuse us.
Okay. In the beginning, like Joanne said, it looked like a stock market, okay? Up, down, up, down, up, down. And when I was hospitalized in 99, for a while, I was on the up and up. And then in 2006, I was hospitalized again for another 2 weeks, received 3 blood transfusions, all the tests. But I do have to say, since 2012, a year after my splenectomy, it's just been up and it's been pretty much level. And I've noticed that I guess I have just the more energy, okay? I have more energy than I used to have. But the one thing that's always, always, always in the back of my mind is because I had the splenectomy. What happens if I dip again? Okay, that's always nagging me. And for some reason, I think that's always going to nag me. Am I going to end up in the hospital again? What are they going to do now? There's nothing left for me to take out, you know? But pretty much that's it. It's been straight. I've been in the normal range since 2012. Okay.
So I agree with Kathy and Joanne. It's been kind of a stock market look. It's definitely not like this. And I never said really how long it took me. It was like 2 years and 8 months to get to this full remission. And I think a lot of what happened too is I was diagnosed before COVID. And every time I get a vaccine, I have a dip. Anytime I get sick, I have a dip, but I'm able to recover. I'm not, some people, have those and then they never come back up. So mine, I'd say, was very much like this, with all kinds of things going along, and then all of a sudden like this. I do know that at some point with CAD, my remission will probably end. For some people, it's 4 years. For some people, it's 15 years. But I am filled with hope. I feel this way. My goal when I was diagnosed, I told my hematologist, my goal is to have a normal HGB. And I was told I will never have a normal HGB. And I did not accept that. Groups like this help with people like Joanne and other people. I was able to advocate for myself and get treatment. And JMO was not approved when I was diagnosed, and it's available now. So that fills me with great hope. And I'm so glad that I've been coming to these meetings. The first time you meet somebody else, you don't know anybody else with AIHI. So it's been a wonderful experience, and I'm very grateful.
Thank you. So for me, it was definitely more of a circle. I would achieve remission, and then I wouldn't. And then I'd try treatment, and so it has been a circle. It has not been linear, and that's okay. And yes, like almost everybody else, I live with the what if. What happens next and what if? But I know that if I have achieved this level of remission for this amount of time, I can do it again. Yes. I can do it again, and no matter what life throws at me, no matter what my hemoglobin dips down to, I have had this amount of time. I can achieve it again. And so I just want to thank everyone on this panel for sharing their story. And I hope that anyone who has not achieved remission yet sees that it is possible. Is it forever? Maybe, maybe not. Everyone's journey is different. But you can go and get symptom-free for some amount of time. And I think we're all open to sharing our stories and talking to anyone, later that, wants to hear more about, whether it's our treatments or whether it's, our, anything else we've talked about. But thank you, Joanne, Kathy, Janelle, and Joe, for sharing your stories. Really appreciate it. And I leave you with a message of hope. With a non-traditional career, with mentors, self-directed studies, and conferences, including Breadloaf twice. Anne is going to be presenting an activity for us that will help us heal and bring out our creativity. Please help me welcome Anne Mallen.
I'm just so thankful to be here, and I wanted to thank Karen and Elizabeth and all all the other presenters, all of you, and just this opportunity to speak to you as a professional when my career was interrupted. I know many of you know what this is like. I was a very young person when I was dealing with endometriosis, had several surgeries, then I had lupus, was diagnosed 25 years ago with lupus, had to stop teaching, and had to reinvent myself several times. So I wanted to tell you that I do understand that. I know several of you have gone through that. I had to go on Social Security disability, have a master's in teaching, but I didn't really get a lot of chances to do what I wanted to do. So I did create a nonprofit. I did a little bit of that. I did a little bit of writing. I tried really hard to find my way through this, but I'm also a breast cancer survivor, and then I got autoimmune hemolytic anemia came first. I had both warm and cold, the monoclonal and the polyclonal IgM and IgG, and then I had the breast cancer. It now looks like it has morphed into a B-cell lymphoma, so I thank you all for your understanding for my need. The doctor said I could come, I could teach, but that I should wear a mask when I'm around the group. So I do really appreciate, of all the people who I could be with professionally, you all understand, and you do get it, so I thank you very much for that. What I'd like to do today is I'm going to share with you a specific tool that you can use. I know a lot of people have said to me, patients that I've met, students that I have taught have said, oh, I'm not creative. I really don't think that's me. I'm not creative. But I will argue with you, and I hope, I'm looking at Joanne, I will hope that by the end of this session, that you will at least understand that, yes, there is something within me that can be creative, even just for myself, for a short moment of reprieve. So I believe that I titled this The Muse Within because it is not just me, but psychologists, philosophers, people throughout history have said that humans are born with the ability within us to be creative, sometimes really creative, sometimes creative in ways that you can make a living, sometimes just planning out where you're going to put the different flowers in your garden. All those things are creative, and I think we forget that. Children know this, of course, but as we grow older, we lose that, and so today we're going to try to get that back. I do want to say very quickly that nothing in this workshop should be construed to take the place of a mental health therapy or treatment. This is just a fun activity. We're going to do a little bit of something. It may bring up some things because, of course, as patients, we are always dealing with mental health issues. You can't not have chronic illness and not have mental health issues. I think that's just part of being human. So none of this is going to take the place of that, of course. And I know you all understand that. So what I thought I would do really quickly is I would just mention a couple more things about myself. You heard my professional bio, and of course, I like to be a little fancy on my professional bio. But what I think would really connect you more to me is if I told you about the little creative things that I do. So, of course, I garden, but I didn't describe to you that I have a mango tree, and then I have pineapples, and then I grow hibiscus and orchids. We have papayas, and we have some other tropical thing I can't remember right now. Bananas. My husband and I cut 2 bunches of bananas, and the gardener is going to get to eat those bananas because we had to come. But those things are really fun, and I take photos, so I'm an amateur photographer, and I also dabble in watercolors. So when me as an artist, or me as a writer in the nonprofit, have a master's and blah, blah, which is more interesting and which is going to connect you? You're going to be connected, I think, personally, to the thing about the gardening and the photography and the parts of me that I'm going to share to you, that you can identify also. We're all like that. We all have our professional selves and we have our creative selves. Sometimes we forget to access those creative selves. So what I wanted to start with is I'm going to go up here to this whiteboard. We're going to do a little brainstorming first, and what I'd like to do is gather all of our ideas about things that you know, either yourself, a creative activity that you do, somebody in your family, somebody you know, somebody you admire. And I'd like to be kind of specific. You could just yell out painting, but it would be great if you said acrylic painting. If you could yell out music, it would be really wonderful. You said, playing the guitar. So I'm going to stand up at the whiteboard. we're going to put all of our IQs together. I have like this much of an IQ. You have this, you have this. We have overlaps of those IQs. But when you put it together in a room, it's massive, right? So let's do that. Journaling or scrapbooking? I'm going to put both as separate line items because you can do that in two different, you can do them together, you can do them separately. Journaling
She
and
said junk journaling.
junk journaling.
Junk journaling.
What is that?
You use scraps and other pieces that people would call trash, and you turn it into a craft.
Yeah.
And you throw it in a There's also the Bible journaling too that people do.
I imagine there's 1,000 ways of doing journaling because as a writer, I know that people sometimes journal, and some people even write their songs from their journals.
Mm-hmm.
They do musical things. We're getting some good stuff.
Creative.
Ah, I love it. I'm a huge fan of the creative writing. That is right up my alley. Short stories, essays, poems, all that fits in there. Memory handbooks, you can read these along with me. But the interesting thing about this is that all of these activities can lower your blood pressure and increase your dopamine, which is, we forget that, right? Those are the kinds of things that we should be thinking about as a patient. You all can hear me when I just yell like this? So we don't really remember, I think, as a patient, that we have tools to lower our blood pressure and raise our dopamine. Maybe some of these things you will think about trying, and maybe there's something in your childhood or something in your earlier years that you might resurrect and decide that by the end of this workshop you might want to get back to. Let me come back to some of my notes, and then, of course, I don't have to yell as much. So we did our little brainstorming activity, and I think it's really important to remember that we don't often utilize this as much as we should. So I made that point. But I want to tell you the story about my mom. My mom was in the last years of her dementia diagnosis, and the doctors had said to her that she probably, well, her and my sister and I, who were her caregivers, that she probably did not want to go through treatment for her CLL. So she had chronic lymphocytic leukemia, but also dementia. So she was going to die of dementia. We knew that. So it didn't make sense to put her through the chemo of the CLL. And that doctor, hematologist, said, but I do have a prescription. I want you to take up a creative expression, anything, but I think you should start a creative expression. And my mom had been a painter for her earlier years and had given that up as she got older. And we took the doctor's advice and we pulled out the pastels. We pulled out her oil paints, her acrylic paints. We pulled out her easel. We got her brushes out. And it wasn't something that she could do at the same level as she used to do, but boy, did it help. It really was something fun for her to pull even old things that she hadn't finished and just kind of finish them up, you know. She could add some colors to it. So that was the first time that I heard a doctor actually say a prescription for creative expression. And I thought it was really interesting because he said very clearly, we have the research and we know that this is beneficial. And so for the purposes of this workshop, I went to go look for the research because he said it's out there. And as a teacher and educator, I knew this was such that even years ago when I was teaching 25, 30 years ago, this was well known in educational psychology. But I'd like you all to go to the handouts that have been given to you. You can put the circle page aside for now. We're going to do that in a little bit. But pick up this little handout that you have. I'm going to highlight a couple things that I think is really good because, well, I wanted to have a little bit of something for everybody. So those of you who are very connected to research, I've got something for you. The people who want some visuals and a craft activity, we got something for you. And people who want to just hear me talk, I don't know how many of those of you, but I got that for you too as well. So right now, I'm going to just point out a couple things on this Journal of Creativity. This is a meta-analysis or a collection of research pieces. I did see that the printout that you have doesn't give you the actual sources because that was in blue ink, and so it didn't print out. However, you can always go online if you want to look this up. You can find this. You'll have the reference if you need to go looking at them. But the author is talking about creative expression has the potential to promote the cognitive, emotional, physical, and social well-being of individuals of all ages. And then he goes on to explain this really cool, on the second page, psychosocial model of creativity and mental health. And there's just 3 things that I want to point out that he's pointing out, and I think they're really important. The first is that any type of creative expression functions as a release valve. So you'll see it under emotional regulation on the second page. He talks about how this helps us when we are stressed. It functions as a release valve. And they know this. They've done many, many studies, and there's all the information about that. You can enter a state of flow when you're doing some artwork. The second thing is that it gives you that kind of cognitive flexibility. When you practice creative expression or do anything that's dealing with expression, creative expression, you get a mental resilience, a little adaptability. Your brain can benefit from the coping skills. And the last thing is the social connection. So I would recommend that whatever you choose to do, you find some way to make it social. And all of these things can be shared with others and can be shared with family, friends, anyone, and it's sometimes even done in groups. So they found with the research that when you're working with others, you actually get a sense of well-being and you get an actual lift. So those 3 things, this kind of release valve, the ability to be a little mentally resilient, and then that social connection, that chance for you to be with other people and to share your work, those things are really important for any type of creative expression. So what I'd like to do right now is just give you a couple quick, and this might be a little bit, this is just my writer self. I can't help it. These are the words. I love words. So we're going to go into a little bit of definitions of what are we talking about when we're talking about creative expression. Is all of those things? Sure. But we want to also think about the definition. Albert Einstein said creativity is intelligence having fun. It's a good definition. Creativity is intelligence having fun. But I would argue that that is missing one little piece that we need, that this particular author, this Jean Berluche Dussel, says we need that additional thing that we need to go above and beyond. And on the first page, he talks about creativity can be defined as a dynamic process whereby an individual or group, through the interplay of skills, cognitive processes, and environmental influences, generates a tangible output. So I personally feel that in his particular research, the tangible outlet is the thing. It's the key to this. You can sit and listen to music, and you can have a moment of daydreaming. You can actually sit and imagine or listen to poetry. Those things are creative because you're actually spending some time with your intelligence having fun. But what I'm recommending, and what I think this author is really pushing, is that in order to get the best benefits, you need to make something, and you need to share that with others. So that's what we're going to be spending a little bit of time. But before we do that, I want to give some credit to the person that really introduced me to this Mandela idea. In 2000, I was newly diagnosed with lupus and going through a divorce, a difficult time in my life. So I definitely needed a release valve. And the local hospital had a workshop for patients who were dealing with a chronic illness or cancer. And so I signed up for this thing, and I went, and we were giving information on mandalas, how to create them, what are the benefits of them. And I thought it was a really interesting thing. I thought that was pretty progressive in 2000. I was talking to Sean. He said Moffitt Cancer Center has been doing a mandala workshop for 25 years. Arts and Medicine program has been in existence for 25 years. So this has been a really big movement in the therapeutic arts part. And I don't think any of our hematologists are actually giving us that prescription, because it's not going to fix or help us get our hemoglobin up, right? It's not that they're going to be spending time on saying you need to do some creative expression. They're going to make sure that we get the treatment we need. I'm just suggesting that we can add this and put it in our pocket as an extra little tool that we could actually use. So that particular nurse, Sharon Cohen, had a master's degree, and I was really impressed with what she did. And over the years, I have created just anytime I felt a little off, a little stressed, a little bit just wanting to do something, I have drawn these mandalas. Some of them you'll see in a moment here. before we go further on that particular slideshow and before we make our own, I'm going to give you the definition. The reason I'm choosing this mandala is it's an easy thing that you can do at any time. I've drawn you a circle. There's markers there. We're going to make our own in a little bit. But before we do that, I'll give you a definition. The word mandala is Sanskrit for circle. And in some references, they actually include the word magic. They'll say magic circle, which I think is a little bit much. But the interesting thing is that throughout human history, there have been multiple groups of people that have used this. The Buddhist monks, the Navajos, their shamans. You'll see them in Christian churches when they're making the Rosetta windows. It's said to symbolize unity and wholeness. Carl Jung believed that mandalas could function as an intermediary between your subconscious and your conscious mind, which I think is really interesting. And perhaps that's where we get the benefit, where we can get an opportunity when any of this creative expression and doodling and making a mandala gives a chance for the subconscious to share what it wants to share. And whether you believe your unconscious or subconscious has anything to tell you, it doesn't really matter. It can simply be just a fun activity. So Elizabeth, now we are going to take a look at some images. And well, before you can get it, you can load it up, and I'm just going to say one thing. Why am I going to show you pictures from cave drawings from way back? What I want to do is I want to link us to humans, all humans. So you'll see the very first one here. That's not it. That is that really great Austin. That's our really great Austin guy. There, that one. That's the oldest known example of a mandala. It's in a cyclone cave in the Kimberley in Australia, 40,000 years old. This image is Colin Murty of News Corp, Australia. I will mention the images that I'm using. I'll give credit to them. If it's in the public domain, I won't say anything, or if it's my photo. So 40,000 years ago, somebody painted that on the roof of a cave. And this next one is Native American Chumash. It's in a cave near Sierra Nevada, California. They think this one is 2,000 years old. I mean, it could just be the sun. This one here is a Jain representation of the, oh no, this is still the sun. This is still the Chumash, Native American one, 2,000 years old. This one, this is a Jain representation of the cosmos. The Jain was an ancient Indian religion. They believed in nonviolence, compassion, and self-discipline. So it's interesting, that's another one of the Jain representations of the cosmos. And now we're going to be looking at Nepal. This is colorized with modern colors, but it's an ancient mandala that's from the 4th century. And then we're back in Navajo territory in our area. Shamans created and still create mandalas for people who are ill and just suffering, and then they have the patient sit on it, and supposedly it absorbs the toxins. If only it were that easy. They will destroy that after they make it. This one is from a temple in Bhutan. It's a Buddhist representation of the creation of the cosmos with orbits of the planets. That image is by Ursula Gansler. And you can see those swirlies. This is a modern Buddhist monks came to our local botanical garden in West Palm Beach to create a sand mandala. This is for compassion for all sentient beings. They worked on this for days, and then when it was over, they destroyed it, as Buddhist monks will do. It's to symbolize the impermanence of all things. And this is like all the symbolism of what was in that mandala. It's really complicated. Now you're going to see some mandalas in nature. This is some pictures from my backyard. I do enjoy photography. I told you about that. And so you'll see many times flowers. Well, even in the greenery, you can find some mandala shapes. You're going to see some of my beloved pineapples here, and you can see the mandala shape. That's the beginning of a pineapple. And then there's some flowers that will give you some of the shapes of mandalas. This is wildflowers in North Carolina. And another pineapple. I think I get a little bit heavy on the pineapples here, but I really love them. And then the last well, it's not the last image. The next image, you're going to see a rose. Okay, that's one more flower. Here comes this was one I did at that first workshop when I was on antidepressants and just diagnosed with lupus. And you can see I didn't really know anything about how I was feeling. I was a little bit stunned. This is when I was going through the divorce. Kind of funny, a little bit contentious. This is when after I had some dental work, and I didn't realize that I was doing needles and teeth until much later when I finished it. This is a springtime when I was feeling much better and things were going well. Several years after I had been under control with the lupus, another springtime, things were going well. I was entering a new part of life. And this is a labyrinth at the Chartres Cathedral in France. And you can walk a mandala. Some people do choose to walk and either meditate or pray. Now you're going to take those circles that you have there on your table, and I want you to not think too much. I want you to just doodle. Whatever you feel is what you want to put into that circle. You've got colored markers. We'll take a few minutes here and we'll just decorate with whatever you like. And when you're finished, then I have a few more things to share with you. I'll give you some ideas of something I would recommend that if you feel like you're finished and you want to do the very next thing, or if you just want to think about doing this next part later, you can take out your little orange notebook, or on the back of your mandala, you can just write down the colors you chose. Just if you chose 3 or 4 colors, just write down those colors. And then, just without thinking too much, write down what you associate with those colors. This is very individual, very specific. There are some researchers that have said what they believe colors are associated with, but I have found that it is very unique and individual to the person. And I will tell you a little bit about what one researcher has thought about with the color association, but you should do your own association. Write down the name of, whatever the color is and what you associate with that color. And you can keep writing if you wish. You can keep decorating your mandala while I tell you a little bit about how you might choose to evaluate your doodle. There's no need to actually evaluate it. You can simply do this for the blood pressure drop and the dopamine rise in dopamine. But if you want to think about what it could mean, Jungians believe the subconscious will speak to your conscious self in the symbolism of color and shape. So Susan Fincher, who wrote a book, Coloring Mandalas for Insight, Healing, and Self-Expression, she has collected some information from multiple sources and says that red often signifies passion, life, love, and the power to create. It can also represent blood. Blue is the color of sky, space, heavens, and eternity. It's often calming and links us with nurturing aspects of life. Yellow is often linked to the sun, of course, and suggests rhythms of time. Sometimes yellow represents the soul of a person, although white can represent the soul as well. Green refers to nature, balance, and harmony, plant fertility, freshness. Orange is often earthiness and contrast to spirituality. Purple for centuries has been associated with nobility, refinement, and power. Turquoise suggests generosity, love, and healing. Traditional people in many parts of the world paint doors and windows turquoise for protection and safe boundaries. Brown is the color of earth, and as an avid gardener, I see it as foundational for energy. White refers to blinding light of spirit, purity, innocence. And black refers to remembrance, eternity, solidity, constancy. You can also consider the structure of your mandala. I'm just going to give you a couple of them. There's a whole lot of information. If you're interested, I did print up a little bit of information about the shapes that you might have chosen. There's a woman named Joan Kellogg who was a sociologist, and she did a lot of work as an art therapist in the 60s, and she studied thousands of mandalas. So she has information on the shape of it. So if you would like that, I have a few handouts that a few copies that I made. But for now, I'll just note that if your mandala has an empty space with a center point, or if you have things swirling inside the circle, You may be at the beginning stages of something new in your life. If you made a cross inside the circle or drew right angles, which Joan Kellogg referred to as squaring the circle, you could be in the middle stages of something and enjoying more independence and competence as you choose some goals. If you found yourself drawing a gate, like a gate-like shape, you might be getting ready to end something and start something new. And that's the perfect place for me to end this presentation on the link between creative expression and well-being. As you created your mandala, I hope it provided you with a bit of release valve and that you see this as an opportunity. And I do hope you might consider looking at things that you've done in your past. And anytime you are in the hospital or you're stuck, you can simply draw a circle. If you have a pen, you can doodle. I didn't have markers, but I just did a little doodling. It looks like I'm in the middle stages, and I think that's probably true of the advocacy work. That may very well be true of what I'm going through. And then I also, of course, since art needs to be shared, I have my own little swag. I made bookmarks for you with paintings of my butterflies, my beloved butterfly. So my husband, Ed, will pass out those bookmarks if you're interested. And I thank you for your time.
Hello, welcome back. My name is Kathy again, and I'm here to introduce the wAIHA Warriors Vice President, Sean Powell. Sean is the Senior Director of Care Management at H. Lee Moffitt Cancer Center in Tampa, Florida. Sean is responsible for administrative oversight of the case management, social work, and chaplaincy care departments, tobacco cessation, arts and medicine, and lodging. Sean has previously served in leadership positions with St. Vincent's Healthcare, New Directions, Behavioral Health, and the American Cancer Society. He has served in clinical roles at the Massey Cancer Center at Virginia Commonwealth University Medical Center and with the American Cancer Society. He earned a Bachelor of Science degree in psychology and a Master of Social Work degree from Radford University. Sean possesses board certifications in oncology, social work, and clinical case management from the Association of Oncology Social Work and the Commission for Case Management Certification, respectively. Sean is a board member of the Board of Oncology Social Work Certification and currently serves as treasurer. Sean's clinical interests include end-of-life care, issues facing long-term cancer survivors, and utilizing technology to improve the patient experience. Sean?
That makes me sound way more fancy than I really am. And I'm going to offer a reward to anyone that kills that damn gnat that has been flying around for the last 2 days. 2 of them? Okay, 2 people get 2 rewards. We'll come up with something. So, and I don't like to stand still. When I'm speaking, I move around, and this is a wobbly stage, so if I fall down, it's not because I had a drink at lunch. Okay, that's better. So I did not come up with this idea for talking about intimacy on my own. I'm nowhere near that creative, as evidenced by the mandala I wrote or drew earlier today. But in December, I was asked to speak at the Colorectal Cancer Alliance annual meeting, which is just like ours. It's for patients and caregivers, but it's for anyone that's been diagnosed with colorectal cancer. And they were asking me to speak on the quality of life issues that people with that diagnosis go through. And 15 minutes before I was getting ready to go on stage, they said, "Oh, and can you talk about sex and intimacy too?" And I'm like, sure, I'll wing it. I'll come up with something. And so I did, and we talked about the physical changes, the effects of chemotherapy, and all of these things. And I did not know how that was going to go over with the audience. But I saw people starting to nod their heads. And then afterwards, and especially that evening at the dinner, I had a number of people coming up to me to thank me for recognizing that intimacy is an important part of their life that was affected by their cancer diagnosis. And that's when I began to think about it and reflect on my experience and how our relation changed by the diagnosis, and I thought, well, let's talk about it. Let's talk about sex, baby, like the 90s song. Jen wanted to play that as I was getting up here. I'm like, no, we're going to keep it classy, nothing like. So, well, now why isn't it advancing? Okay, there we go. So I'm trained as a professor, which Karen will appreciate. So we always have to tell you what we're going to tell you before we tell you it. So it's objectives, and that hopefully by the end of this talk, you'll be able to define what intimacy is in the context of a romantic or a personal relationship, understand the issues we face during and after treatment, and then identify new ways and also expectations of intimacy in your life going forward. So as we get into this, how would you all define intimacy? Yes, physical touch. Spending time with your significant other. Quality time. Okay, it's quality. I heard somebody over here. Oh, the feeling, the emotions that go with being close to someone. Well, the definition we're going to use for today, first, we all know what wAIHA is, warm autoimmune hemolytic anemia. But intimacy I took from Merriam-Webster. It's something of a personal or private nature, closeness, both emotional and physical. Because a lot of times people immediately, when you hear the word intimacy, you think of sexual intercourse. And intimacy takes on so many different things. It can be that feeling of closeness, the feeling of love, the feeling of commitment. Intimacy is a wide-ranging emotion. So what are some normal expressions of intimacy? And you all should probably be very familiar with it. It could be sitting close to someone like we do in a movie, or cuddling up on the couch together at night to watch a TV show or binge something on Netflix. Physical touch. I'm seeing a couple over here touching each other on the shoulder, touching an arm just out of a desire for that feeling on your skin. Kissing, hugging. Of course, sex also plays into that. It can also be exchanging notes and letters. We do that on holidays such as Valentine's Day, birthdays, Christmas, also Hanukkah. Sharing deeply felt emotions. I remember when I wrote the vows for my wedding, it was easy to write them, but when I got up there to say them, I started bawling like a little kid. I couldn't get through it because of all that emotion that was coming up with me. And also that feeling of trust and safety. When we're with someone we love, we feel safe. We feel like if something happens, at least I've got this person with me, and they're going to be in it with me, and they're going to help protect me. So this, relationships are complicated, but this intimacy is what brings us value, and it's what keeps us committed to the individual that we're with. So how does wAIHA affect intimacy? So obviously, once you're diagnosed or as you're going to the doctor trying to figure out what the hell's wrong with you, your focus is really on surviving and figuring out what's wrong. Because I know we've all, as we were going through that, you were afraid, am I dying? Am I going to get through this? And that really takes precedence over everything else in your life. And we experience changes in roles and relationships. As I've listened to so many people's stories, Jen talked this morning about having to give up her job, and that impacts you financially. She had a role that helped identify her, and it changed in her relationship. You may experience anxiety or depression, leading to a loss of interest. That anxiety can come from the shortness of breath. It can come from not knowing what's wrong with you. The depression can come from lack of ability to do things that you normally want to do. The grief from losing your health. Like for me, I'd been healthy my entire life. I had just run a half marathon and woke up on a Monday and something was wrong with me. You also have few chances to be alone during the treatment process. I was in the hospital for 2 weeks. I was not alone for 2 weeks. And as somebody who likes to spend time alone, that was the most miserable aspect of being in the hospital. So there's really, logistically, there's few times if you're undergoing treatments or you're in the hospital for any type of sexual intimacy. And then there's dealing with the side effects of the treatment itself. And I did a little research on this, and I found that prednisone can actually reduce sexual desire. Plus, anybody who's been on it, you really don't feel like having sex because you feel like you're going crazy. And also with it, it comes the physical changes to your body. the weight gain, the moon face, and then you don't necessarily feel as attractive, so you may not be interested. We talked about the financial challenges and then the shortness of breath. Sex makes you short of breath anyway because your heart's racing, but our hearts are racing due to the disease, so you really don't feel like it. And also the fever, the chills, the rigors, you feel really awful. I did read a study that says that anemia lowers testosterone, and both men and women get their sexual desire from the hormone testosterone. So if your testosterone level is low due to the anemia, you're not going to have that natural hormone to help you feel interested in sex. And then just all of the different changes that go on in your body that affects your feelings of self-confidence and your feelings of being physically attractive to another one. You got the weight gain. Some of us have talked about an altered body odor. When we were actively hemolyzing, we didn't smell the same, and it was a very strong, unpleasant odor. You may have a PICC line coming out of your arm, which makes it difficult. Some of us talked about the pain, the abdominal pain from the enlarged spleen. You just didn't feel like having sex. And you could have nausea. You may have swelling and then pallor. If you're really, really white, you may not want to take your clothes off. So it's realistic. I live in Florida. We're all tan all the time. If I was really white, I don't know how I would feel about that. So we know that that's how it affects. And I always like to give people some things to take back with them. And so first thing is, what should you talk to your doctor about after wAIHA and sex and while you're on treatment? And Dr. Irina, please fill in if you have anything else you want to add as far as the questions go. But you can ask, will my treatment affect my ability to have sex? And if so, how long will it last? That was not something I ever talked to my doctor about. And really now when I reflect back, we should have had that conversation. Is it safe to have sex? I learned while doing research for this presentation that if you're on CellCept, you should not get pregnant. And I don't know if anyone in the room was told that, but we do have women in here of childbearing age. You absolutely cannot get pregnant while on CellCept. So that should be a conversation your doctor's having with you about if you do want to have sex, taking the appropriate precautions to keep from getting pregnant. Which leads us to, what do I need to know about protection from STDs? We have weakened immune systems, which make us more susceptible. We think of colds and viruses, but also to sexually transmitted diseases. And you don't hear much about chlamydia or syphilis, but those are still out there. And if you are non-monogamous, you do need to be using protection if you're on any type of immunosuppressant. And what are some options to prevent or prepare for sexual health during treatment? What should I be doing? Which is some of the things that we just went over. So what can you do to help regain intimacy after this diagnosis and you're in the healing process? First and foremost is give yourself patience and time. I know that after I got out of the hospital, it was quite a while before I was comfortable with my body again. I had lost over 20 pounds. And for somebody who's already fairly thin to begin with, I looked like a skeleton. And I was gray. And it took me a while before I could regain my color. And I wanted to get back in the gym and feel like my real body again. So first and foremost, give yourself patience. But it can start with something as simple as writing love notes, simple messages in an email, a text message, or even maybe a sticky note. And this is a great opportunity to show how much you appreciate the help that your care partner or your husband, wife, significant other showed during your journey. My now husband slept on the couch in my hospital room every single night. And I wrote that into my vows just to show him, and I'm getting a little emotional now, how much I appreciated that and why that made my decision to marry him even easier to make. So really recognizing that our care partners are going through that journey with us. Set time alone to be together, date night, go out to your favorite restaurant, maybe go for a walk in the park, go to a movie, something you two are doing together. You could even go back and redo your first date, but anything that allows you two to spend time together. I want to leave a few minutes for questions. You can just lie naked together. Sex does not always have to be penetration or anything like that. It can just be the act of lying without your clothes on, next to each other, caressing, spending time together, just physically being present with touch. Enjoying that meal together, taking a walk, reading a book, or you could try something new. We learned today how to do mandalas, so maybe you and your partner do mandalas together or something. When I was walking around this afternoon before this presentation, I saw there's a community chalkboard over there. And I saw a couple, they were drawing on the chalkboard, and I thought that was so nice. There's also community musical instruments out there too, if you want to take a walk. Also, that's set time alone, lie naked. I think this is just a second slide. Also alcohol. So we all know that alcohol lowers inhibitions. So if you are allowed to have alcohol or you do drink, one drink may actually make you feel a little bit more comfortable. Not two, but one. It reduces our inhibitions and can make you feel a little bit more desirable. And if you're comfortable, you can also try new forms of intimacy, spicing things up a bit, touching, kissing. Obviously, there's a lot of toys. You don't need to go to a store. You can get them on Amazon now, so there's privacy. And there's some wild things out there as I was looking for this talk. And then some I didn't know what you did with. But, and you can also ask your provider for a referral to a sex therapist. I don't like psychology today because you can go out on psychology today and just say, I specialize in this, this, and this. And whenever I see someone that is specialized in everything from CBT to sex therapy to trauma therapy, I'm like, no, you didn't. No, you didn't. You can't go to school that long. So I would talk to your doctor. They usually keep a list of good therapists that they recommend. I would also say you could go to the National Association of Social Workers. They have a referral function as well to find a qualified. And you can always go to your state board. Like in the state of Florida, you can look up all the social workers who are licensed and actually see if there's any discipline on their record too, because you don't want to go to somebody who's gotten in trouble. So with that, I wanted to give you some resources. Obviously, wAIHA Warriors is your best resource. There's also the National Association of Rare Diseases, the Autoimmune Association, and a new one I just learned of, which is the Global Autoimmune Institute. So I wanted to leave time for questions.
Our next session is called How to Proclamation with James Romano, and I'm going to read his bio for you. James Romano is the founder of Care and Cure Partners. Care and Cure Partners works to advance the advocacy, nonprofit fundraising, and government relations needs of clients. James has over 25 years experience working with nonprofit organizations in a government relations and advocacy capacity. Throughout his eventful career, James has worked for a member of Congress, nonprofit organizations, and Washington, D.C. lobbying firm. James brings his experience to the benefit of the Care and Cure Partners clients. He began his career in patient advocacy at the age of 19. Maybe you should tell this story. As a freshman in college, James skipped class and went to Capitol Hill, where he advocated for the passage of the Ricky Ray Hemophilia Relief Fund Act on behalf of the hemophilia community and his family members with blood clotting disorder. Because of his family members with rare and chronic conditions, James devoted his professional life to advancing public to removing barriers to access of needed health care. In 2005, James became the Director of Government Relations and Advocacy at Patient Services Incorporated. From that position, James built relationships with multiple state and federal policymakers, as well as nonprofit organizations. James focused on issues involving health insurance coverage and access to treatments and therapies, including promoting the charitable assistance model. James is an expert on patient assistance issues and can guide patient advocacy groups through that process. James and Care and Cure Partners assist in building advocacy communities and advancing agenda and legislative goals for organizations. We've been working with James and Care and Cure Partners for a while now, and he has been our host on Warriors in the Hill for the last 2 years. And so today he's going to talk to us about proclamations.
I'm going to be speaking to you about, I'm calling it state advocacy, proclamations, art acts, how we can build campaigns on a statewide basis to really bring awareness to wAIHA, AHA, and how you can work together, how you can actually do it in your community. So thank you very much for allowing me to speak. Actually, I thought I'd start out with something fun, the 10th Amendment to the Constitution. The 10th Amendment says that it reserves the powers for that are not specifically delegated to the federal government by the Constitution and are not prohibited to the states respectively, or to the people. So in the United States, we have federalism, we have the federal government, we also have state governments. And beyond that, I had a college professor who always says, anytime we needed something, we just throw a layer of government on it. So you have towns, cities, county government, school boards. So you have all types of government for different things. And every time we needed something, okay, let's just throw a layer of government on there. And so, and usually I've been always focused mostly in my career about Congress. I like to think of myself as a person who's pretty expert on how to deal with strategies on dealing with the U.S. House of Representatives. But actually, what we're going to talk about today is more and more how you can engage your local officials and your state officials. And I think there's a role for each and every one of you moving forward to really promote wAIHA and AHA and CAD on these levels. And I'm looking forward to any questions you may have on that, but this is where I would recommend. Proclamations. This is a great campaign to raise awareness for wAIHA, for AHA, or any disorder, really rare disorder, that people don't know about, and it gives you the power. So this past March 25th and every March 25th is AHA Awareness Day. And I know in the future, we're going to want next year for you guys to reach out to your states and in your local governments and ask them to pass basically a proclamation recognizing the 25th as AHA Awareness Day. Now, you're probably thinking to yourself, well, how am I going to do that? First of all, I don't know how to do it. And believe me, I didn't know how to do it either, but you learn how to do it. Why do you want to do this? Like, why do I want to engage my county commissioner or the mayor of my town? Because on a grassroots level, this is how you raise awareness for your disorder. This is how you support the advocacy of the organization. And if everybody in this room does it, then it really does amplify the voice and start really bringing recognition to wAIHA and AHA. To our partners in the room who are manufacturers, this gets them to show off that wAIHA is actually working on something and working on something very important, and how they could be a part of it too. So the biggest question is, how do you get a proclamation? And it's a great question, because if you've been working with one state, you've been working with one state. All 50 states are different. They all have different requirements. They all have different time periods. They all have different, I'm going to move forward real quick. This happens to be the information for the city of Austin and how to get a recognition for your different disease and putting together a proclamation just for the city of Austin. And so, as you can see here, they have, they give you the different things they offer. They give you the different timelines. This is who to contact if you want to come pick it up. I work with the Bleeding Disorders Association of South Carolina, and March is always Bleeding Disorders Awareness Month. And we put in the paperwork, and the governor of South Carolina always, for the last 15 years, has done a proclamation honoring March as Bleeding Disorders Awareness Month, which was recognized originally in 1981 by President Ronald Reagan. And when we go do our advocacy day at the state capitol, we meet with the lieutenant governor, a very nice lady named Pam Yvette, who then reads the proclamation to us. We all take a picture, and this has happened now for 15 years. Okay, so what did we get out of this for 15 years? Well, when we have a problem with public policy, then she sits down with us and talks it and calls the governor, calls the speaker. So we've built that relationship by getting this proclamation that has spread out to other groups and to other issues that we're working on. So it's very important. So you can contact your county commissioner, the mayor of your town, and ask the process to be able to obtain a proclamation, either for AIHA Awareness Day, or if there's ever a wAIHA month. We've never did. So when I first started working with Karen, we were like, what would be a great month for wAIHA Awareness Month, or week or day? And I know at one point we were looking at December, and they were like, oh, okay, that's not going to work because it's December. Yeah. Okay, but you can work with once you start obtaining the information, you can work with the wAIHA warriors to get the right information that you want. I do want to go back here though. Usually it's about a 60 to 90 day contact that you have to fill out the paperwork and get it. And if you're right on the cusp, they're not going to give it to you. It's very you do have to be a little bit more pragmatic and get out, or not pragmatic. Get out in front of it so people can't, so that these states can do it. Because even if you do fill out the paperwork, sometimes they don't send it to you. I mean, it's just, it gets lost in their bureaucratic shuffle. The farther you go with governors, with mayors and county commissioners, then sometimes not. But the good thing is, what I've seen over the years is if your town council then decides to make March Hemolytic Awareness Month, they'll invite you to the meeting where they vote, and they'll give you the proclamation. You can say and take pictures with people. So it is also a great social media opportunity. It's a great, like I said, raising awareness opportunity. So where are you going to find this information? Honestly, you can Google the governor's office for your state, and then call, and that you can find, and they'll put you in the right direction. My wife's boss is the county commissioner she's a teacher, and the principal of her school is also the county commissioner. And so my wife emailed her and says, my husband wants to know how we can designate a certain in the county for AIHA Awareness Day. And she gave me the email me, tell me what you want, and I'll present it to the Board of Supervisors. So it could be like the Board of Supervisors all the way up to the governor of the state. And so that's an important thing. So that's how you can obtain your proclamations. And I think it's great as a community that's, we're in our third year of having these meetings, that this is so important that it's something you can do. It's something you can reach out to on your day off, or on during the day. You can, at your lunch hour, you can look up who some of the important people are in your town and your state, and you can work with your town council or the mayor's office, because this is something they want to look positively to. They put out we sent out 50 proclamations in the last year when they're running for re-election. So it's a win-win situation for everybody. And I hope it's something when we send out information next year asking you to do this, that you'll help us, and we'll engage with your local governments and the governor's office in your state. And what we're going to do is, I'm under the impression that probably in January of 2026, we'll start putting out a process for everybody if this is what they want to do. So this was the information that actually when I worked with Karen this year to submit to Ohio and Virginia. These are the whereas, it's general information. It's not whereas we're the greatest, disease disease community to ever come to pass, which I think you guys are. But, this is to give information about the disorder, and the more information, and you want to keep it concise. You want to make sure it fits on one page, and get it signed by the mayor or the governor or the town council. And then what's the greatest part of that is generally speaking, the lower the government that lower level of government you're at, the more likely you'll be able to participate. And like I said, take pictures and be able to use it, and then send those to Karen, and she'll publish them hopefully in the newsletter and on the website. This is a great opportunity to raise awareness for you as a patient advocate and leader, for the organization, for the disorder. So it's something that is very important. Just to show you, just if you haven't seen, Karen got to meet the former speaker last year in Washington. We were there for an event, and I just like that photo. Oh, that wasn't me. Yeah, during our first advocacy day, a group of our patients, anybody, Justin's not here, Eric's not here. They actually got to, like, they exited an elevator, and there she was. And it was like, oh my God. And they took pictures with her, engaged with her, so it was great. And then last April, we got Karen to be able to engage with her. So it's, it's very good. And actually had a kind of an opposite story like that. A couple about last week, a person who had never been to Capitol Hill before had to use the men's room, and he goes into the men's room, and as he's coming out, Marjorie Taylor Greene was coming, and he like knocks right into her, almost knocked her down, and I was like, oh, good God, that's all we needed. And he kept saying, I didn't realize how short she was, and I was like, yeah. I said, you almost knocked her down. He goes, oh, I'm sorry, I didn't, but it was just kind of a funny thing. Proclamations is one form of something to raise awareness for. Actually, Dane and I work with a group that not only do they try to get a proclamation from every governor, but they see what different landmarks they can get lit up. So this is a tower in Atlanta that they were able to light up orange for Chronic Disease Day 2 years ago. And you can work in your town and ask, okay, what can we light up orange? I guess orange is our color for wAIHA, or for AIHA Awareness Day and AIHA Awareness Month. And they'll give you a list, and you probably will have to fill out some paperwork and do some follow-up. But that day they can get it lit up. And so you'll go downtown that day, and they'll light up the bridge, and you get a picture next to it, and you'll send it to Karen. Last couple weeks ago was the first day. It was International Periodic Paralysis Day, and Dane and I work with that group, and they had the Chicago skyline lit up purple, which was really cool. And they were able to, it was beautiful at night with the purple. And so why I'm saying this is, it's more teal than purple, but this is something you can also do while you're getting your proclamation. What can we light up the color of orange? And then it'll be say, oh, we're lighting up the tower today for Hemolytic Awareness Month. And so it's something to consider an awareness campaign with on the future. This other picture is a State Representative Stu Jones of Lawrence, South Carolina, and he is recognizing the hemophilia community in South Carolina during their annual Advocacy Day last year in March. And a lot of his colleagues stood up behind them as he's doing this. This is something that you could do with your state representative or state senator. You can go to them and ask them to recognize the community on that date in your state capital, and it'll be in their record, and then you get a copy of the record and frame it. And it's something that you can do. Now, we were there in person, but it's something that you can ask them to do without you being there, or you could go to your state capital that day. Karen's not that far from Columbus, but it's something that you can then start working. And I always say it's very important that your local reps, whether it's your state representative, your state senator, that you know who they are and they know who you are. I tell this very funny story. I was in Walmart, and people know me. I have 3 daughters. I'm a proud father of 3 daughters. My youngest 2 are twins. They're the cutest things in the world, except when they're monsters. And my wife thought it was a great idea if the 5 of us go like shopping at Walmart while the kids were like 4 years old, or 3 years old, and they're in the cart, and we're going down the cereal aisle, and they're knocking the Fruit Loops and the Lucky Charms, and finally exasperated, I said, would you put the effing cereal back? Just then, my delegate from the house taps me on the shoulder and says, hey, Jim, how you doing? So a couple months later, I'm at an event with him, and he goes, "You should see Jim yelling at his kids in the Walmart over the Fruit Loops." He was saying the F word and everything. And I was like, but he remembers me now, and he remembers my children. And but what I'm saying is if you learn who they are, and you start developing a relationship, and I hate to say the lower that they are, but like it's your state representative. They'll usually sometimes they'll come to your house to meet with you, or they'll meet with their local office, or you meet you at a coffee shop. They really much actually want to talk to you and learn about your disorder and see if there's anything they can do for you. And you can say, can you do a statement on the floor of the legislature? Or can you invite us? And that'll help. Can you help us get a proclamation from the governor? We've tried. We filled out the paperwork and nothing's happened. Can you help us? And that's something that they're going to be like, sure, that's something easy for them to help you with. So, so it's something like we can do that moving forward in the next years to help you reach out and talk with your local representatives about wAIHA. Another thing I want to let you know is states are, there's been NORD, Sean mentioned NORD, which is the National Organization of Rare Disorders. And they've had this campaign over the last couple of years to put together a rare disease advisory council in every state. As you see, they've been very successful. Local advocates like yourselves on a local level have gone to state capitals and gotten bills passed creating these councils. And actually, Kathy and I were talking. She's going to go advocate on Rare Disease Day in the New Jersey Capitol, she was telling me. And I said, well, they have a rare disease council. You should talk to your representative about trying to, next time a advocate, a position opens up, to have her state rep or state senator nominate her to be a patient representative on these state commissions. They don't pay anything, so don't think, but this is something that they bring you, you meet every couple times a year and you make recommendations on how to promote rare diseases in your state. I'm going to tell you that with the climate now, and Dane's going to talk after me about the climate now in DC, I think a lot of things are going to go to the state level, and especially on rare diseases, your voice would be having the wAIHA voice sit on some of these panels is not a bad idea, again. So I just wanted to point that out to people as a way of promoting. We can promote through proclamations. We can promote through meetings and having statements about wAIHA and AHA in the General Assemblies. We can promote awareness on the disease by getting involved with these legislative rare disease advisory councils. I know the head of the Ohio one, and I've sent her like 50 emails and copied Karen, trying to get Karen nominated. So one of these years, we're going to get Karen nominated. Okay, so people always say to me, well, how do I know who my state legislator is? Well, as I said, if you've been to one state capital, you've been to one state capital. If you've been to one state capital website, you've been to one state capital website. Arose by any other name. For some reason, when it comes to the House of Representatives or the lower house of state government, they go by different names. And I think it has to do a lot with how it developed, even from colonial times. So using, Kathy as an example, New Jersey, they're called freeholders. I don't know how that, in some places, in New York, they're assemblymen. In Florida, they're representatives. In Virginia, they're delegates. So, but it's the same person. It's your person who's sitting in the lower house, lower chamber of the state legislature. 49 states have what is called a bicameral legislature. We get this from English common law and English history. There was the House of Commons and the House of Lords. When the United States was set up in the Constitutional Convention, they wanted to have two houses like the Lords and the Commons. They called it the House of Representatives and the Senate. And then states said, okay, we'll have a House and we'll have a Senate. One state in the Union, is there anybody here from Nebraska? You're the only state that has a unicameral legislature. So, and they all call themselves senators because I think that's a better title, I guess. So that's, and then in some states like Maryland, New Hampshire, you have a senator, and then you actually have multiple delegates. So you have a couple people representing you, which is great if you're trying to get co-sponsors on a bill. So, because they usually vote for, they want to help their constituents. So here's South Carolina's. You can see in the bottom right, or bottom left, excuse me, like you could put in your address, your zip, and then it comes up with who all your elected officials are. You look at your state senator, you look at your state rep, and then you can click on them and go and request a meeting, or you might know them. You might went to high school with them, or your kids play ball together. So it's something to reach out to, and then to introduce yourself and talk about wAIHA and how it affects your life. Here's some examples of state advocacy. The gentleman I told you about who saw me at Walmart, that's him with my wife and my baby and my goddaughter. And then that little baby is the one in the middle with Senator Norawaki and Representative Cabela from Hawaii. And the picture down there is in Pennsylvania. That's Representative Abigail Salisbury and most of a lot of the Democratic caucus in the Pennsylvania House of Representatives. Here's some more. This is Massachusetts Rare Disease Council. This is a representative from New Mexico. Why I really do believe getting to know your state reps, your state senators is going to be very helpful in the future. Plus, you never know when they're going to be your next. I'll tell you this funny story. My dad had a friend who was a state rep in Pennsylvania named Larry Curry. Larry Curry had been there like from time, I think, from time to beginning. And he was retiring, he shared this room with this other guy, this other state rep who was a freshman. And he said, you should really get to know my suite mate. I think he's going places. And so I went and introduced myself and said, it was nice to meet you. And he goes, yeah, hi, I'm Josh Shapiro. If you guys know, Josh Shapiro is now the governor of Pennsylvania and one of the front runners for the Democratic nomination for president in a couple of years. And there he was, a state representative who I met, whose father was a hematologist and who championed standards of care for hemophilia patients in Pennsylvania. Why I say that is you never know where your state rep or state senator is going. Some of them just might be sitting there for 50 years. Some might end up as president of the United States. Look at Barack Obama, he was a state senator. Build your relationships now, and it'll help the community in the future. So get your proclamations, work with your town councils, work with the governor's office, work with your state reps and state senators, and ask them to help you get these proclamations. Send them on to Karen, and she will decorate a wall at her office with them. But the point thing is that it's a piece of paper, and that's great. What you're getting is they're going to know you. So in a couple of years, you're going to be at Walmart, and they're going to say, okay, there's Karen with wAIHA. And that's what we want. That's what we want. And then they're thinking, well, I'm about to vote on Medicaid cuts or expansion or something. Let me ask my friend Karen, who I know who comes in here, what she thinks of it and how it's going to affect her community. And that's where we want to get. So when the big issues happen, they know to come to you because they know your story. They know that you're living with a chronic, rare disorder, and they'll take your story under advisement. That is the underlying reason we want to do this. So I probably went over my time, but does anybody have any questions?
He's the managing partner of the Health and Medicine Council, has worked in government relations since 2004. He specializes in advancing medical research, treatment development, and patient access-related policy goals for a variety of stakeholders. He has a background in grassroots organizing and healthcare advocacy, as well as a detailed understanding of the federal budget and appropriations process. Dane holds a Bachelor of Science in Political Science with a concentration in economics from Marietta College. He also holds a certificate in leadership from the Bernard P. McDonough Center for Leadership and Business Studies and an MBA in multi-sector healthcare management for St. George's University. And it's turning it over to you, Dane.
Thank you, Sean. Is the sound coming through okay? Excellent. There's another, I think Sean made some excellent words about sharing your story. and the power of your voice and the importance of feeling like you're being heard and that you're comfortable. And there's another audience for that as well. And that's what we're going to talk about a little bit today. And that's policymakers and your members of Congress. We don't need to share in the same way that we're sharing here. But if there's one thing that your members of Congress understand, it's that you have a pure self-interest. They hear from lots of different stakeholders. They hear from the insurance industry. They hear from PBMs. But they want to know how the system is working for you. You can tell them that better than anyone else. Surprised that there weren't more therapies available. Surprise at things like out-of-pocket costs or insurance practices. This is what they need to know. And if you don't tell them, there's a good chance that they wouldn't be familiar with it. And it's particularly important if you have a rare condition, because there's always that issue of, if I'm not the one telling my story, who else is? It might not be the same situation for something like COPD, but when you have a situation where you have 435 members of Congress, you have 100 senators, if you're not making your voice heard, who is? And if nobody is, then how would they know? How would they know what the patient experience is like? How would they know how they can help? So I do know there's an awful lot going on in Washington right now, so I do want to leave enough time for questions at the end, but really quickly, I thought this was an excellent introduction. My name is Dane Christansen, managing partner at the Health and Medicine Council. We are a nonpartisan firm. We specialize in something called the appropriations process. You might be familiar with the golden rule. Whoever has the gold makes the rules. That's what appropriators do. They assign federal funding on an annual basis. Great way to make policy. You don't have to pass bills every Congress, but you have to pass spending bills every year or the government shuts down. So an awful lot gets done in the legislative process, but also in the appropriations process. The Health and Medicine Council works with many different patient organizations. These range from very large groups like the American Lung Association to smaller groups like pulmonary hypertension and the rare disease community. And I mention that because there's always a journey. Everybody's looking for the same thing. It starts with research, breakthroughs, scientific advancement. It goes to treatment development, improvements in patient care. Then it's coverage and access. Once those products are coming to market, how do you use them effectively? How does your doctor know what they are? And then there's an important public health component of that too. Are we doing surveillance and tracking? Professional education, public awareness? All of this touches the government. And when you look at a condition like pulmonary hypertension, there was a time about 20 years ago when your average life expectancy was about 2.5 years from diagnosis. Today, that's well over 20 years. You always hear things like, geez, we cured polio, but what else have we done? The truth is they make incremental progress time and again. Wonderful scientists like we have in the room are probably better at discussing this than I am, but it's one foot in front of the other. It's a drop in the bucket, but over time that bucket fills up. You start to see pretty significant advancements, and of course you hit a tipping point where a lot starts to happen. I'm going to start. How many folks in the room have advocated on Capitol Hill? Done a meeting in DC? Sent an email? Excellent. Maybe done a local visit? Well, all right, maybe I'll get more hands on the second question. How many folks have been in 7th grade before? All right. Probably learned civics or government at that time. Some very simple and fundamental things. We had a revolution about 250 years ago. We decided we didn't want to have a king. We said we're going to set up a democracy. We're going to have separation of powers, 3 co-equal branches of government. Legislators which make the laws, courts which interpret the laws and decide balls and strikes, an executive branch that enforces the laws. All of this is the same today as it was when the founding fathers started to set up. Things have changed, but the framework and the process are the same as they ever have been. I mention that because I know you turn the news on, it seems like everything is just going crazy. A lot of this is the same as we learned it in 7th and 8th grade. One of those things is true, Article 1 of the Constitution. Jim got Article 10, I'm gonna get Article 1. Congress has the power of the purse. Taxpayer money can only be drawn from the Treasury by Congress. House representatives and the Senate are the ones that decide how much money, where it goes, how it gets used. The administration has no grant-making authority. They can't design programs. They can't draw money out and decide how to spend it. Only Congress can do that. There's a way to do things. I had a friend who just became a naturalized citizen. Called me up, he said, I'm so glad I'm getting naturalized. You should probably be worried though, I heard birthright citizenship is going away. I said, what are you talking about? He said, didn't you see the news? The president signed this order. There's no more birthright citizenship. And I said, well, that's, not all executive orders are created equal. They're great to have that photo op at the desk with the pen, but the reality is if you want to change how citizenship works, 3/5 of the states have to amend the Constitution. Jim, is that correct? Yes. All right. Jim's the expert on all things founding fathers related and the business of the country. You might be a little bit more familiar with something even more recent. There was an announcement that came out about defunding PBS and NPR. And PBS and NPR were nice enough to release a statement that said, we can't be defunded, we're not part of the federal government, and the government doesn't decide how we spend our money. It's a long-winded way of saying the news is there for a reason, but don't believe everything you see on TV every day. And don't forget, your members of Congress work for you. And they make these decisions based off of your input. They care what the White House says, but they care a lot more what their constituents say. That's how the system is set up. The courts aren't designed to take your input. They're designed to read the law and say what's right and what's wrong. The executive branch is there to simply implement what Congress has enacted. Congress is passing laws, deciding how much money goes where, what they should do. They rely on input from you to do that. They want to know what their constituents, the people who vote for them, think is important, and they want to have that information. There's one other important thing, I think, though, that was in the Constitution and how everything was set up, and that's free speech. And this might come as a shock to you, but you can say almost anything. You can't do almost anything, but you can say almost anything. And a lot of people in DC don't tell the truth all the time. They might just lie to you. So once again, I think it's an important distinction to note what's being talked about and what's being done are not always the same thing. But the important thing to remember is the process and the significance of engaging with your members of Congress. So with that being said, I'm going to tee up just what's happening in Washington right now, bring you up to speed since we started back in 1776 and are catching up to 2025. Right now, today, we have a new Congress and a new administration. President Trump was the first person since Grover Cleveland to be elected to 2 non-consecutive terms. He was the 45th president. Now he's the 47th president. But that also means you got a little bit of a preview of what kind of president he's going to be like 8 years ago, before the previous administration. That administration took office on January 20th. One of the first things that happened was a number of executive orders. There was an executive order that came out freezing all federal funding. This executive order took Congress by surprise. There were a bunch of members of Congress, including Republicans, that were at an event at Mar-a-Lago, started getting calls from their governor, started getting calls from defense contractors saying, what is this? Are we not getting our money? They had to call the White House, and in a couple of hours, the White House was saying, oh, big misunderstanding. We're not freezing all federal funding. We're just going to freeze some federal funding. But there's the squeaky wheel gets the grease component, which I want to remind everybody of. And there was even an executive order on something called the F&A rate, or indirect costs for research, which was probably a little more esoteric, but I think it's an important encapsulation of how things work and how they tend to work within the system. The administration said, we're going to change how indirect costs work. So if I think people know, research money comes from the government, but the money doesn't stay in the government. It goes to Baylor, it goes to Moffitt, it goes out to scientists all across the country who compete for it, and it funds their academic centers. And they get something called indirect costs. Indirect costs is how they do things like pay for folks to clean the lab, how they get lab mice. These are indirect costs. It's not the science itself, but it's the infrastructure that comes along with it. The announcement said, we're going to change how this is funded. We're going to take that rate from 60% down to 15%. Huge disruption for research and would change how things are getting done in this country. There was an injunction put on that almost immediately, within hours, so it didn't go into effect. And then, of course, if you're a judge, you have to make a decision. Can the administration change this policy, or is it up to Congress to decide what the F&A rate is? Well, if you're a judge, this one wasn't terribly tough. You probably make a lot of tough calls. This one, in the law, in the literal law that was passed on March 14th and has been passed, this was the annual spending bill for about 10 years previously, Congress sets that rate, and they even include a sentence that says, and the administration can't change it. So if you're a judge, you have a pretty easy call to make here. So the administration on appeal withdrew that challenge. The FNA rate isn't being changed, but we do expect they released something called a budget request to Congress. This just came out a couple of days ago, where they say, Congress, this is what we would like you to do. So remember, Congress has to make the laws. Rest assured, saying change that FNA rate is going to be one of those things they ask Congress to do. Congress, though, I think as people know, does things in a bipartisan and bicameral way. It's not a tug of war, which is why the same hot-button issues get debated over and over again. They never get solved because they can't be solved. When you disagree, the system doesn't move forward. It only moves forward where they have common ground and where they find agreement. They're not going to have common ground on changing how research is funded, so that's very unlikely to move forward this year. We have some new agency leadership. Anytime we have a new administration, you usually get new leadership of new agencies. This includes the National Institutes of Health. Technically speaking, if you're an agency head, when the new president starts, your job has ended. You are summarily dismissed unless they proactively reach out to keep you on. So this means all of our key healthcare agencies, the National Institutes of Health, which does research, the FDA, which does drug review and approval, CMS, which covers Medicare, Medicaid, private insurance, the CDC, which does public health, all have new leadership. The Senate spent a lot of their time early in the year confirming these new nominees. They don't tend to confirm people based on their political ideology. They confirm them based on whether or not they think they can do the job. You can say what you want about Dr. Oz, but Dr. Oz is also a very reputable surgeon from Columbia. So many of these folks, the new NIH leadership, the new FDA leadership, CMS, have the medical credentials you would expect them to have. Increasingly, though, they will be taking over the decision-making role. What's getting done in terms of research? What's getting done at the FDA? This will migrate to the director level and away from the White House as you have these new leaders confirmed. One of the things that occurred more recently that caused a lot of consternation in headlines was this, what they call the RIF, or Reduction in Force and Reorganization, that came out at HHS. So they announced on April 1st, many people thought it was a joke because it was April Fool's Day, but it was not. They said they wanted to reduce about 20% of the staff. This is something that is within the executive branch's power to do, and they said, we're going to reduce these employees. NIH lost about 10% of its workforce. Other agencies lost more. This is something that Congress, though, has not been happy about. When this RIF and reorganization was announced, the reorganization is something that Congress has to do. I'm going to point to a different executive order for that. Some folks might remember that one of the first executive orders had to do with the Department of Education. And it said we're going to end the Department of Education, and then it had another line in there that said, to the extent allowable by law, which means to the extent allowable by law is that Congress has to pass a law ending it. So Congress is very unlikely to do that. But similarly, if you're going to reorganize different HHS agencies, Congress has a role to play there. But the who gets fired is a different story. Congress is not happy about the staff reductions, and that's why almost immediately the HHS secretary was summoned to Capitol Hill to testify. They're still working that out. There's been some closed-door briefings, but it's safe to say that, if you think about those three separate co-equal branches of government, Congress does not feel like their guidance is being followed, or they at least want to know, well, if you're reducing staff, then what are you doing to make sure all of the money we've provided and all these core functions are still doing what they're supposed to do for what we assigned each dollar amount for? And then, of course, more recently, there's some issues with tariffs. And you might say, well, geez, why are we even mentioning tariffs in a meeting like this? Because tariffs cover medical devices, generic drugs, healthcare as well. I didn't know what to say for this one specifically because it was a pretty safe bet that tariffs would be on again, off again, up again, down again. But traditionally, medical devices and products have been excluded from tariffs. Not this time around, and we're going to have to see what that situation is, but there's mounting pressure and public outcry that we can't have things like generic drugs coming in and facing some absurd tariff just because they came from one country or another. So this is something that we're working on now, and a very large stakeholder cohort is pressuring members of Congress and the administration to deal with this issue. So what does that mean for the House and Senate, our new members of Congress? They took office on January 3rd. They've been around for a little bit longer. This was a big election in November, though. We already knew a lot of folks were retiring or not returning. This was going to be a sea change election regardless of which party ended up in power. There's roughly 70 new members of the House of Representatives out of 435 members, there's 10 new senators. So 10% of the Senate is completely new. But the key thing was the people that left were not folks who lost in a swing district, pendulum swung back. They were not new folks that just got there. It was a real change in leadership. Kathy McMorris Rogers, the longtime head of the Energy and Commerce Committee from Washington, decided she was retiring. Kay Granger from Fort Worth, Texas, longtime appropriator, said, I'm leaving. Dr. Michael Burgess, one of the House healthcare leaders, also from Texas, said, I'm done too. So now, as Jim noted, we have a bunch of new folks, and we also have people that we might know and be friendly with us that are elevating themselves into leadership positions. They're the new folks writing these bills. They're the new folks deciding what gets done. Very important that we maintain connections with them and make sure they know who we are. We wouldn't want to have a good relationship with Kay Granger, the old appropriations chair, and now Tom Cole from Oklahoma, the new guy, doesn't know us very well at all. We want to make sure all these members know who we are and know how they can represent the community. Which brings us to the appropriations bills. Fiscal year 25 is the current fiscal year we're in. It started October 1st of last year. Congress tries to finish the spending bills by the end of the calendar year. They almost never finish them by October 1st when they're due. They love to procrastinate. Last year, the last Congress, the 118th Congress, didn't do a lot of work. They didn't pass a lot of bills in regular order. They had this great idea, kind of like cramming for one of your finals, at school, that they were going to ram everything through at the last minute. They loaded up a spending bill, and then they added all the bills that they hadn't done to it that normally would pass just in a typical 2 years. This included things like Medicare physician reimbursement. They always fix the rate at which Medicare pays doctors. The rate goes down, harder for Medicare beneficiaries to find a doctor to see. They need to plug the Medicare physician reimbursement, 2.8% that was being lost. They needed to reauthorize something called the Pandemic All Hazards Preparedness Act. They needed to reauthorize the Priority Review Voucher Program for rare diseases at FDA. And they needed to do things like pass a bill called the Kids Access to Care Act, which said if you're in the Medicaid program, you can see a doctor across state lines if it's specialist and things like that. They loaded all this up, and then the package fell apart at the last minute. So none of these bills prevailed. They punted into March 14th. The new Congress had to dispense with this, and on March 14th, they dealt with this the simplest way they could. They said, we're just going to keep all federal spending exactly the same as it was last year. They froze everything in place at the FY24 level. This was not a bad thing. It was okay. Could have been worse, but they'd forego some opportunities that were there. The Senate had proposed spending bills that had more money for NIH, had more money for CDC pending. They also had $250 million for what they call flagship initiatives at the FDA. This is or at the NIH. This is the Brain Initiative, the Cancer Moonshot. They had extra money for that. And they also had money for the DoD Congressionally Directed Medical Research Program. The DoD program only does work where Congress says work in this area, but it's funded because the House provides half the money, the Senate provides half the money, and then they merge it all together in something called conference. They didn't get to do that conference, so they didn't get to merge all their money together. So about half the money was lost for the program. So the same amount of work to do, but only half the budget to do it with. These were the negative outcomes. The administration said they want to change, how HHS operates, but they have to abide by each of those funding lines. If Congress has provided $10 million for the blood disorders program at CDC, they have to provide $10 million for the blood disorders program at CDC. And also some of these things, some of these executive orders are still facing legal challenges, what grants can be frozen, what's happening. All of that's percolating through the courts right now. You probably saw the Supreme Court ruled about the whole issue with the wAIHA, saying that the wAIHA is appropriated and authorized by Congress. It has to keep going. So a lot of that stuff is percolating as well, but leading to a very dynamic environment. But the important thing is you want those spending bills to have your money included and to have the guidance that you want included as well. So what are the community priorities specifically in any given funding year? Fund medical research. The high tide raises all boats. The more money they provide for NIH, the more money there's available for any specific condition within NIH. Heart, Lung, Blood Institute at NIH is someone we work very closely with, and also they have the Division of Blood Disorders. NCATS, the National Center for Advancing Translational Sciences, houses the Office of Rare Disease Research and something called the Rare Disease Clinical Research Network Consortia. If they have more money, they can fund more science. And if you think science is overfunded, the current pay lines previously were about 15, 16%. So they're not funding every science. They're not even funding half the good ideas. They're funding more like 1 in 8 of the good ideas. More money gets us to better numbers. The CDC has something called the Chronic Disease Education and Awareness Program, or the CDEA. The CDC puts out announcements and says, bring us your best ideas and we'll fund you. We'd love to be funded by the CDEA, but of course, they need more money to fund more projects. The Senate had a $2 million increase pending for the CDEA, but then they got level funded, so the same amount of money. Much harder to do new activities. The blood disorders program continues to be very important. This is what funds things like the hemophilia treatment centers. It also funds all rare blood disorders work. It's funded about $10 million. We'd love to see that number go up. A lot of these programs have had increased funding for years. The CDC activities have not. That needs to change. The VA has a very active research program, and the DoD, as I mentioned, has a very active research program, including something called the peer-reviewed medical research program, which we want to be part of. We want to say, you need 2 senators to put you in the DoD program to compete for that funding. We want to have that critical mass of support so we can compete for funding as well. We were trying this year, and we also need them to restore full funding for the program. If it was a normal year, they would have the whole $1.6 billion that we were talking about, not the $800 million that they ended up with. So they need to restore all that money. They need to put us in the program. This is a member-driven process. Members are driven by their constituents, and they're going to add that money because we asked them to. If you think there's an awful lot going on with executive orders and everything with the White House, the Congressional Black Caucus Foundation has a very nice executive order tracker. If you're wondering where anything is at, you can just go to it and click on it, and it will give you all the stuff that's been done and what its legal status is and where it's at. Highly recommend it. It's a great resource if you're trying to keep tabs on any of this stuff. But aside from appropriations, what's happening with the 119th Congress with patient care legislation? I'm sure folks remember Schoolhouse Rock. Not just bill passes the House, passes the Senate, gets signed by the president, but also if you don't make it through in 2 years, you have to start the process over again. So a lot of these bills reach a tipping point. It's very unusual for something to happen in Congress and then get funded or get done within one Congress, that first 2 years it's there. Usually support builds up over time until it hits that critical mass. And the example I usually go with is something called the Genetic Information Non-Discrimination Act. Anybody would say this is common sense. What Gina said was, if you get genetic testing done and find out you have a predisposition for breast cancer or colon cancer or anything else, then that information can't be used against you. A life insurance company can't deny you a policy, and your employer can't fire you because they say, oh, you might cost us more money down the road. We want to know your health status. It took 10 years to pass that bill. But when that bill passed, it passed 420 to 3. So once you get over that level of support, you kind of break through and your time has come. So a few bills that are close that the community's been working on. The first has to do with prior authorization, a doctor's prior auth. That prior auth bill had 350 co-sponsors in the last Congress. In any normal 2 years, it would be done. We need to get it over the goal line this year. There's a bill called the Safe Step Act, which has to do with step therapy or fail first protocols when some insurance company says you got to use a cheaper product and not what your doctor said. All that bill says is your doctor gets to know if this is happening, and you get to know if this is happening. They're switching you onto something else. And if this is a serious condition, your doctor can overrule that. They can say, I'm the expert, you're not the expert, this is the product we're using instead. There's also a bill called the Help Copays Act, which has to do with copayment assistance, and says if you're getting copayment assistance, it has to be counted to your out-of-pocket maximum. An insurance company can't double dip by taking the copayment assistance and then saying, oh, but you haven't reached your maximum either, so keep giving us money until you hit that cap. Congress is going to address PBM reform, or pharmacy benefit managers. I think this, everyone knows, has been a key discussion. There's an open question, which was they allowed it all this vertical integration, the doctors, the insurer, the specialty pharmacy, but the whole idea was it was supposed to save money. And Congress is saying we did this and it costs more now, so what's happening? So they're taking a look at this. They haven't figured out exactly what they're going to do, but they do want to address what's called cost shifting in that, which is when they push the cost back onto patients as a way to blunt them utilizing their health care. They say you got to pay more and more out of pocket because they know, particularly if you have a rare condition or a difficult one, the more expense that falls on you, the more likely you are to drop out entirely or end up in Medicare, Medicaid, or a different federal program and not on the private insurance programs. Physician reimbursement is going to have to be dealt with. The kids' access to care is going to have to be dealt with. And the FDA's peer-reviewed voucher program will have to be dealt with too. It still needs to be reauthorized. Just because they didn't do it last year doesn't mean they can't do it. They have to do it. So really quickly, the Help Copays Act is another bill that's getting close to that tipping point. It has 150 co-sponsors. It's led by a guy named Buddy Carter from Georgia, who's a pharmacist. The reason that the bill hasn't been introduced yet is that Buddy Carter has now become chairman of the subcommittee. So chairmen typically don't drop bills. They need a new congressperson to step up and take over that initiative. And in the Senate, the bill was led by Roger Marshall, who's a doctor from Kansas. 25 senators, but that number can grow. This bill has only been around for about a year, though, or 2 years, though. So this is a newer bill, already amassing about 142 co-sponsors. Safe Step Act's been around a little bit longer. The maps mean that the states have dealt with this on the state level. We're asking for action on the federal level. What type of health coverage you have shouldn't be determined by what state you live in. There needs to be what would establish is a minimum set of standards for everybody. So everyone have a basic level of protection. A state can go beyond that, but nobody can be excluded from that. In terms of the Safe Step Act, it was led by a congressman named Brad Wenstrup. Brad Wenstrup retired, so same deal. A new person has to step up. But that bill had almost 250 co-sponsors. In the Senate, the bill had almost 50 co-sponsors, so right at that tipping point of majority support. It went over majority support in the House. It's right at majority support in the Senate. Just a little bit more needed to get it done. And then what's going to happen looking forward? There's going to be what just happened, which is a budget request for fiscal year 26. A lot of the stuff the administration wants to do, saving money, consolidating agencies, they have to send to Congress to do. They can't just do it themselves. That budget request just came out. It's non-binding. I want everyone to understand that. You're going to be a lot of headlines that say the administration cuts funding. They haven't cut anything. They've proposed. They've asked Congress to cut the funding. Congress is probably not going to do that because they hear from their constituents that say, we need more money for research. We need more money for the FDA. But that's the budget request that's coming out. They will decide this through the FY26 appropriations process. Just because it took them extra time to finish FY25 doesn't mean they get more time to do FY26. They still have to get things going by October 1st. And this is when they're going to decide whether or not they put conditions like us in the DoD research program, how much money they give to the Heart, Lung, Blood Institute, how much money they give to the CDEA, maybe to do even more projects with us. The one thing you're going to hear about a lot, though, which is another reason why all these bills haven't been reintroduced just yet, is something called budget reconciliation. Budget reconciliation deals with entitlement spending, so not necessarily NIH and the CDC, but the bigger programs like Medicare and Medicaid, and it has to do with taxes. What are we collecting in taxes, and what are we dedicating those funds to? There's an awful lot going on right now in budget reconciliation. You're going to hear about 3 things moving forward, particularly in the House. One is going to be student loans and kind of big changes to student loan programs to generate savings. The next is going to be SNAP, Supplemental Nutrition Assistance, the food stamp program, which is going to be just potentially talked about this week. And then Medicaid, potential changes to Medicaid. Of course, you can say objectively or in general, we'd be against cuts to Medicaid, but it's always easier to say that when a specific proposal is on the table. You can reject something Congress has put forward rather than just kind of an idea in principle. They have to package all this up together. They can't pick and choose each one. They can't just do student loans or just do the Medicaid piece. It's all or nothing, and they're on a ticking clock to get it done by October 1st. The House has taken kind of a very draconian path. The Senate has taken a more moderate and incremental path, shifting some money to defense, to border security, to traditional conservative priorities, but not the way that the House has that requires them to cut all these other programs. They have to figure out how they're going to square this circle by October 1st. They need the House and Senate to pass these bills. But you're going to hear an awful lot about this, and it's going to delay action on some of the other things like help copays and the Safe Step Act. But moving into the August recess, when we'll be gearing up for advocacy again, they'll be working on those bills during the second half of the year. Still need to pass them. They can't forget about them. And then there was already going to be an effort on Capitol Hill to decide if we need to modernize the NIH and the CDC. Every few years, Congress decides what these agencies look like or what they should do. That was already coming up. The key person that's going to be playing a role in that is a guy named Bill Cassidy, who's a gastroenterologist from Louisiana. He is the head of the HELP committee in the Senate. His partner is Bernie Sanders, who I'm sure folks know from Vermont. They're going to be crafting the bills about what NIH should be doing and what the CDC should be doing. So once again, this is a process that's going to go through Congress. Very much want them to have your input or information and know what they could do for our communities. The most important thing to remember, though, is that just tell your story. You don't have to get involved in, all this work is nonpartisan. It has nothing to do with Democrats or Republicans. It simply has to do with telling your story. So all you need them to know is who you are, where you're from, what the communities you represent, and why it was important enough for you to talk to them. That matters a great deal as they make these decisions. You need them to co-sponsor the Help Co-Pays Act. You need them to co-sponsor the Safe Step Act. You want them to put us in the DoD peer-reviewed medical research program. All you have to do is reach out and ask. Jim and I can help with this. We'll be working with Karen on this, but telling your story is very important. We also can use your stories. If you have a testimonial or a statement, Sean's been nice enough to assist with this in the past, we can include that in the materials that go to Capitol Hill. Whether it's testimony, fact sheets, other things, we want them to know what the community is all about. I assure you, there's more staffers that say, what is wAIHA, than say, oh, I know what's happening. There's about 100 folks that are what they call AAAS fellows. They're from the American Academy for the Advancement of Science. There's some other Robert Wood Johnson fellows. Basically, every other staffer is someone who came from the campaign or is just looking for a job. Those folks probably don't know an awful lot about healthcare in general. They certainly probably don't know about our community. We want them to have that information. There's numerous opportunities to do this. Meeting in DC, meeting locally. Every member's office has local offices. And of course, phone calls work, but emails work even better. You can reach out to them. You can say, I just wanted to give you this information. Here's what we need you to do. And of course, I don't want to filibuster the entire thing. I do want to leave enough time for questions. So since we got about 8 minutes right now, happy to respond to anything anyone is wondering about, please. This is the question is, there was a decision made recently called the Chevron decision, which curtailed the ability of agencies to do things. You know, we said, like, a good way to think about this is when the court is calling balls and strikes, like with that F&A or indirect cost topic, they're looking at what's a gray area. If there's a gray area, the administration can step in and make policy. They have ways to do this, rulemaking, things like this. But if the letter of the law is clear, the administration does not have much latitude. What the Chevron decision said was it pulled things even further away from the administration and to Congress, and said you can't just run wild whenever there's no clear law. You have to follow as closely as you can what Congress is intending for you to do. So this is going to put more onus on actually your senators and representatives to do two things. One is pass more bills to be more prescriptive. Instead of just saying like, hey, why don't we start a new program on clean fuel? They're going to say this is what that program should do and here's how it should work. So they're going to pass more bills, and then they're going to pass bills that are much more detailed. So they're going to have to come up whenever they're doing something, like the next time they pass the pediatric priority review voucher program, the bill might go from 4 pages to 8 pages because they're going to say, and we want to get into the specifics of how this is going to work. We don't just want to leave it up to an agency that might do something completely different with it. Yes, it's going to make it much easier for rules that the government is creating, not Congress, but the administration, to be struck down in court. But if Congress is passing laws that are detailed and numerous, then the law is what will prevail. So this is more about the rulemaking that the agencies would be doing, where the law is unclear. But that's why it's always important to talk to your senators and representatives. There's a couple things that I think we want to be sure we're clear on when it comes to effective advocacy. We want you to know who the health LA is that works for your member of Congress, and we want them to know you. So you're not filling out a form on the website that says, tell the congressman what you think, or congresswoman. Put this information in the field, and maybe in a couple weeks you're going to get a form letter back that says, thank you for contacting me. Your opinion is very important. That email went to an intern who assigned it at, You want to email the health LA directly and say, hey, it's me. We need you to co-sponsor the Help Copays Act, or we need your support to include us in the PRMRP. That is very effective. When you hear about things like calling, it's usually when there's a tug of war going on. Congress is going to vote on potential cuts for Medicaid. They want as many people reaching out to the office as possible so they can say, oh, there's 20 calls that came in the pro column and 10 that came in the con column. That's important, but most important for us is making sure they know who we are, what we do, and the specific things we are asking about. We float on this higher tide, but we have to make sure we're advocating for our own specific stuff. Even entities like NORD and EveryLife aren't advocating for wAIHA-specific priorities. Only we are advocating for wAIHA-specific priorities. But don't send a letter, though. Letters do not, they have enhanced security on the Hill. If you send them a letter or thank you note, it shows up about 2 months later, it gets irradiated in Ohio. Don't send handwritten correspondence, as nice as it might be. The Health Legislative Assistant. Every office has one. We would want you emailing them directly. And of course, at a certain point, we want them meeting with you, even knowing who you are, establishing that relationship. Yep. Jim, do you want to comment on that at all, or you want me to take the lead? Okay. There's a couple things I'd mention. One is the Help Copays Act, or copayment assistance, is part of this. The community, in addition to passing the bill, you always want the force of law, as we just talked about, but the community also sued the government a few years ago. They sued HHS for allowing copay accumulators to even exist. And they said, this is wrong. And they won that lawsuit, and they won on appeal. They're supposed to be writing new rules right now that would allow anybody to access copayment and other forms of assistance from nonprofit or other sources. The other thing that I would mention that's taking place is that there's been a number of bills introduced that would enhance the Americans with Disabilities Act. So the ADA protections are constantly being looked at, and this includes air travel, other stuff. That is something Congress is keenly aware of and needs to be modernized as well. And there's another part of that that's had a number of bills too, and it's the disability process through Medicare. The idea that it takes 2 years to qualify, or you have to, people understand that's just too long. The system doesn't work. If you're supposed to be out of work for 2 years and getting on SSDI and then how are you supposed to do this when you have a rare condition? So there have been a couple of bills introduced that are going to overhaul the disability fast track process, or allow people to qualify for benefits much more quickly, rather than just saying, oh, you got to be out of work for 2 years and then get SSDI and then get Medicare. So that one, the Help Copays piece, and these are all bipartisan. I wouldn't imply that it's one party or the other, or who's in charge matters. These are all bipartisan initiatives. It's simply elevating them to that level of attention where the Congress says this is what we need to work on next. So I'll give my honest opinion. Tom Cole, who writes the House bills, is a shrewd politician. He was even considered for Speaker. I think he has a difficult caucus to maintain. I think people know recently the dynamic had been that about the Democrats and half the House of Representatives had been carrying all the bills forward. This had been true for the past 3 years. Whenever they had something they had to get done, even though it was a Republican-controlled House, it was Democratic votes and half the Republican caucus. So Tom Cole in the House is a tough job. He has half the Republican, the two halves of the Republican Party they disagree more than they agree. So he's always been good about trying to placate everybody. And what that usually means is that they adopt a lot of things that the administration puts out. They agree to the budget request stuff in one manner or another, or some of these policy changes, but they don't fight for them behind closed doors. And they include a bunch of what's called report language, like including folks in the DoD program, because that's the House component. The Senate has always written bipartisan bills. The Senate has a long history of doing things together, Democrats and Republicans, and that we'll see if that is maintained again this year. But the bills traditionally do include modest increases for the NIH, for the CDC. The bill that didn't pass previously, I think, had a $2 billion NIH increase that was pending, in addition to that extra $250 million. The Senate may do something very similar again this year. And then it's up to the Senate and House to figure out what that compromise package looks like. And, we know Trump from 45. He's going to stake out his priorities ahead of time, but he doesn't veto a lot of bills. Most of the time, if the Congress is sending something through, he says, okay, And I think that if the Senate and House reach agreement and they have a bill that has modest increases, I think it's going to get passed into law. The other alternative is going to be that they just do another CR. They say it's too hard and they keep everything level funded again, which of course ends up being a cut when you factor in like inflation. But even in that scenario, they can do things like add us to the DoD program. It doesn't cost any money to put us in the DoD research program. So they can still make progress on our initiatives. But I think the two options, I don't think there's an option that says we're cutting any research funding. I don't believe Congress would vote for that. I think it's either level funding if it's too hard for them to reach agreement, or modest increase if they say this is where we want to go with all this. So thank you very much.
So I have been very humbled to have conversations with many of you. I'm Ann Leone. I'm the Global Medical Affairs Leader at Johnson & Johnson. And hearing your personal stories over last year and over today and yesterday has really touched me. And so last night, my husband and I on the phone worked together to write a poem that we call A Tribute to the Journey of wAIHA Warriors. In the shadows, weary souls tread. Tired hearts, heavy with burdens unsaid. Warm autoimmune hemolytic anemias call. A battle fought daily, yet we stand tall. Fatigue wraps like a blanket heavy and tight, yet a spark burns within, igniting the fight. For in the journey, we're never alone. A community rises where compassion is shown. Warriors united, advocates of change. With knowledge and care, we help to arrange a tapestry woven with stories and grace, sharing experiences. Each finds a safe space. A personal touch, engaging and real, providing comfort that helps all to heal. Empowered by purpose, each voice speaks aloud. In this embrace, united, to stand strong and proud. Through research, we can reach for the stars to transform the struggle, heal the scars. Together we rally with hope as our guide. Support that is strong, a community wide. Here's to the wAIHA warriors, brave hearts intertwined, advocates facing challenges, resilient and kind. With collaboration and love, rising each day for brighter tomorrows, leading the way. Thank you very much for the opportunity to share this with you. It comes from my heart.